So good morning to everyone. We'd like to start the second day of the Brazil-France Health Workshop. I'm sorry, now it's so I'm going to start again. Good morning. I'm going to start. My name is Cristina. I'm an ID physician. I'm professor at Escola Paulista de Medicina, Federal University of São Paulo. My research line is in antimicrobial resistance, and I would like to start today, the second day of the Brazil-France Health Workshop. Thank you very much for your presence, and I hope to have a very good day for everybody. So I'd like to start asking Professor Ester Sabino to talk. Ester, I think that she's very well known. She doesn't have any presentations, but I'm going to introduce her anyway. She's a full professor of the Department of Pathology of College of Medicine of the University of São Paulo. She's a member of the Brazilian Academy of Science, and she has received many awards from society, from the São Paulo and Brazilian Society, to her contribution to the Brazilian society. So, Esther, thank you very much for your presence, and please start your talk.
Thank you very much for the invitation. And do I present? And I continue? Yes. Okay. So I thought of showing and giving an overview of what we do. It's not showing there. So instead of just, I changed a lot the things that I do over time, and mainly because of this project. So I thought of giving you an overview of what I have been working, and actually also the importance of this kind of program that we are trying to build now with France, and how when you have a long-term studies in collaboration with different groups, this may go through ways that we didn't think originally. But it's not. Isso aqui não tá bom, viu? Não vai, tá vendo? Ele tá se mexendo sozinho. So I was trained in the US through this program in HLBI, REDS, and through a Fogart program in the early 90s. And this program is now almost 40 years, a program developed to study blood safety. And in 2006, this was open for international sites, and that's when we compete. And what was interesting in this program is that we could go through totally different projects, like Chagas disease, dengue, sickle cell. And they actually, because we start there, we could later on get other grants, like was the case of Chagas with the MRC grant, the dengue that end up in our CADI project with FAPESP, or even sickle cell, that we receive a follow-up grant from TopMed. But what was, I think I'm not going to be talking about sickle cell, but there is a group of people working that, and we're very interested in collaborating if there is groups interested in working with sickle cell. Anyway, this is a study in Brazil that has five hemocenters. I was a PI with Mike Bush. Now we have replacements, Carla Ginardo and Brian Custer. And this program started early, and I was very lucky to have the help of these people who work in computer science since 2006 to help us develop a way to bring in data from all these sites and organize the data. So today we have 4 million data from donations over these years, which help us understand the evolution of several diseases. Here I just add the original, the first studies, but now we can follow them through a long period of time. And many other questions that are helping now the students for different areas of Brazil to go and just have an idea and use this data set for research. The other thing, as I mentioned, was Chagas disease. So Chagas disease is caused by this parasite, Trypanosoma cruzi, that it's transmitted by a bug. And the problem with the disease, and to do the studies on that takes a long time for people to develop the two more severe forms of the disease, that the cardiac form and the digest form. So it really depends on long-term commitment of support. So we were very lucky that we started with REDS, then we got this grant. And here is the things we've been working We are now in the third run of the Tropical Medicine Research Center, and in the first time we focus on the rate of disease progression, biomarker of disease progression. Then we also work with tools such as evolution of Chagas cardiomyopathy, and more recently we are focused on self-cure and Chagas treatment, and also thinking of ways of improve access to care and work with implementation science in a region that's more affected. So to do all this, we had to develop the capacity to establish several cohorts. And these are the cohorts we have and the visits. So you can see there's a large cohorts and with several measurements that can help other researchers that are interested in Chagas disease to work together and bring their ideas, because as I mentioned, we always work in this capacity of establishing data sets, organized data sets, with the support of the group of the computer science at USP. So these are the kind of studies we could develop, like a long-term incidence and predictors of progression of Chagas disease. And also, because you have all these samples collected, we are able to do GWAS studies and try to understand markers of the associated with cardiomyopathy. And this is my colleague, Antônio Ribeiro, who is the co-PI from University of Minas Gerais. So this is a study that's done, the Semitrop means São Paulo and Minas Gerais study. So the others I mentioned to you, and a lot has to do with the interest of US. So somehow the Chagas study, and they were interested to understand the disease because they were about to start screening for Chagas disease in their blood supply. So there is, the support comes with some both interest from both groups. In the case of dengue, the same thing happened because dengue was starting to occur in Puerto Rico, where they do have a transfusion service. And I think now this problem will come to also to France as dengue start to increase there. But in any event, this study was to try to understand what was the impact of dengue in transfusion medicine. And this was a huge study. We had to collect more than 40,000 samples in a very short period of time, donors and recipients, more than 30,000 not testing, and we end up with 120 dengue positives, not positives among the donors. So the key thing was to understand rates of transmission, disease among recipients, but also we could for the first time understand how this can help the public health system. And because we had this, and this was a FAPESP grant, we could sequence those viruses. We started a collaboration with a group from Oxford. They at the time they were collaborators from the US. So we started doing connections that we were not thinking before, but that's how we end up collaborating with a group from Oxford. And one thing that was key, interesting for me, and the Americans had all these, they went through the West Nile virus among the transfusion and understand the epidemic of West Nile virus. So we used something that they have used in this study, which was to test blood donors before and after the epidemic. And by doing so, we could infer how many people got infected in that epidemic in the city. In this case, in 2012, that was dengue 4, we reached the conclusion that about 6% of the population had got infected during that season. So this was for me something, well, maybe we can always use the blood bank to infer what's going on during an epidemic. And after the dengue, there was the Zika epidemic, and now we are changing gears because then the group from England, they knew us and they came and and we learned about this new technology at that time that was nanopore that they have used during the Ebola epidemic, and we started this project that was supported by MRC. It was a Zebra project, and we learned about the technology, established technology in our site. And this was a very important study that we could understand when the virus was introduced in Brazil. So this technique and this study led us to get now a large grant That was the CADE project here in Brazil that had the support from MRC and FAPESP. So now we are out of reds, just moving for new things. And this was studied with 3 universities from UK and different groups in Brazil. And the study started in March 2019. And by December, we organized a workshop. So exactly when the COVID epidemic was starting in China, we had this workshop discussing how to do sequence very rapidly during an epidemic, and we were preparing ourselves to do dengue sequencing. That was the goal at that time. But it changed, so all these people, it's an interesting picture. Most of the people who were working on virus sequencing were in this workshop, especially the group from England. So there is no way that we could not do the first studies in Brazil. Everything was there. The money had just arrived, the money had trained the people. And once the samples arrived, we could do the first sequencing. And this was really, in Brazil, gave a lot of buzz in the media. But also, we were one of the first countries that were able to analyze the first sequence and describe how the virus was spreading in the country. This is a paper on Science early on. I think we submit by June. We had already been able to collect all this data and to publish. So this joint capacity was key for us to do this. The other thing is, because we had this in our mind that blood banks could help, we started a prevalence study on different sites of Brazil. As I said, there were 5 centers. We had some other 3 centers. We could get some local grants now. At that time was Trump, and he was with no money from US, outside of US doing Trump area. So, but we are lucky to have support. So by August 2020, we knew that in Manaus, we had a very big epidemic, more than 60%. I don't know if you remember, in the second semester of 2020, everybody was thinking about passports, who had already got COVID. So there was a big thing on here to understand what was the size epidemic could reach, and especially if we would have reinfection. At that time, most studies were saying that reinfection was a rare event. So we thought of establishing Manaus a cohort to understand reinfection, and we would start this cohort by January 2021, but exactly when the epidemic started. So we were there, and because of that, we could detect the first SARS-CoV-2, the gamma variants, and linking all these things, modeling this blood bank and the sequencing capacity. You could have several papers just describing how this epidemic, how this new variant was moving in Brazil. So the other thing is the fact that this epidemic happened the way it happened very quickly. There was this people were afraid that Brazil was unable to detect new variants. And there was this group from Grupo Mulheres do Brasil and a company, Magalu, that asked us whether we could help and establish a network that at least one city in each state of Brazil would be able to do sequencing and detect new variants. So it was interesting because our institution, Institute of Tropical Medicine, and the Department of Infectious Disease, we have a lot of contacts with many people in Brazil. So one was talking to the other, and we slowly find them, not so slowly, because I think we really were able to establish this. In green are the places where we could link someone to these networks, a laboratory, and we support the Mandelics. Slowly we train one in each site. And by the end, we had more than 60 people trained, and the first Omicron paper came from a group that was part of this network at Federal School in Minas Gerais, Federal University. So it was kind of interesting how you can link, and it's not that difficult, though Brazil looks so large. There are groups that can be rapid, we can rapidly establish this capacity. And I would say that in so for part of this group, for example, after the training was done, they were able to submit a proposal for Amazon 10 Plus from FAPESP. So there are these things, they continue in a different area. I'm not the PI anymore, but they were able to continue that. One of the things that our group are very interesting is developing technologies that can be applied for rapid detection of new viruses. This is one of my PhD students and her work. She helped developing this. Was a collaboration, the Birmingham part of CAD project, and I was used to detect Sabia virus, Mama arenavirus here in São Paulo, and also when the monkeypox arrived, we were able to rapid provide the sequence. As you can see, using this protocol, you may be able to have a result in less than 1 day. So the other thing that I also work, and I think it's important, is that during the epidemic early on, this group from a city, nearby city in São Paulo, right, almost a neighbor, it's in the metropolitan region of São Paulo. They wanted us at the Institute of Tropical Medicine to establish the diagnostic PCR for them early on the epidemic. So instead of doing just the PCR, we start discussing with them whether we should not establish a platform that would control the full epidemic in primary care. And because we had established all these cohorts before, and we always use systems to do that, we could easily provide them with a platform that would help the people. They could call, they could enter the website and give their symptoms, and a physician. So it was like a telemedicine, but all the steps control, including the PCR testing. And the idea started in March 2nd. We developed it very fast and trained the people. And by April 8th, we started the project. And I think this was important. And just to give an idea how it worked really nice in the city. But as of June 23, we were able to just submit and publish data from primary care. So we could collect, when you organize the system to provide care, but you organize in a way that the data is easily extracted for research, this really facilitates the capacity to do it fast and give a response. And I think this is one of the very first studies on primary care data on the symptoms of the people. So that just to show this is where we work almost 3 years with this platform in the city, and that is another example that's showing that the gamma variant. Here we are comparing case fatality, case hospitalization rate, and in hospital fatality rate for different variants, and you can easily show that the gamma was much worse than the others. And this made me think a lot, and the group of like, how can we work new epidemics and use the local, our data sets from the cities. And here is an example that we are using the medical chart to just counting the words in the medical chart may give us some results and tell us what are going on in the city. So this is supported by Instituto Todos pela Saúde, and we can see that the word fever goes well with the COVID. So now we are working to see how can we use these data sets routine and that you can get in 1 day. Just by analyzing artificial intelligence, we may be able to provide evidence of new epidemics. The fact that we had established all this effort in São Caetano, bring in a colleague, this is Ana Levin, she does studies on antimicrobial optimization, and she chose São Caetano exactly because we can easily collect data from this city, and this can be used for other projects there. So this is an implementation science study, and we also have a part that's related to environmental impact and to understand the the local wastewater and the concentration of antibiotics in that area. So we do the intervention. I want to know how this impacted the environment. So these are things that we are very interested here now, starting from now, and just call the attention, just to make the link with France, that Institute of Tropical Medicine is part of the Gabriel network with Meouche Foundation, and we are also working with them this kind of study using water environment. Here we linked with two other countries, Cameroon and Madagascar. They have several 16 countries in the world, and we can link to all these groups through these networks. Quite interesting, and a way that we think is very important. So there were many people working on all this program. I couldn't put all the names on everybody, so just to few pictures saying, well, we were happy doing that. That was a funny time. And of course, acknowledge the support of many agents. Thank you.
Thank you very much, Esther, for the nice presentation. It's very nice to see how collaboration works. And now it's my pleasure to introduce Professor Renato Costa Monteiro Filho. Renato is Brazilian. He was trained in Botucatu, in the Medical College of UNIFESP, Botucatu, but she went for his master's and PhD in France, and now she's chief of Department of Immunology of Hospital Bichat, the Universidade de Paris-Cité, and he'll giving us a talk, please.
Thank you all. Obrigado pela introdução. Prazer estar aqui. It's a pleasure to be here, and thank you for the invitation. So I am immunologist, as Ana Cristina mentioned. So can I have the first slides, please? So I'm not epidemiologist, as Ana Cristina mentioned. I am immunologist and nephrologist. So my approach for infectious disease is through the immunology. So I'm going to present in two parts of my talk. The first part will be immune evasion through immune receptors, and the second part will be the dysbiosis of microbiota that conducts to autoimmunity. I still don't have the slide. It's probably coming, I guess. So a little technical problem. So.
Renato.
Okay, so can you put them? Okay, thank you. So I'm just to introduce my center, director of the Center of of Inflammation in the north of Paris, at the Bichat Hospital. It belongs to the Université Paris-Cité. And also our unity has about 300 people, is 11 to 12 teams, different topics, and concern inflammatory diseases. So My particular team works on immunoregulation for many years, and basically to understand cells like macrophage, for example, innate cells, how they can inhibit or activate the immune system. You see here that our question was mainly devoted to immunoglobulins. So since I left Brazil in '82, I've been working with IgA, immunoglobulin A, and also IgG. And so we've been, during my postdoc, I was lucky to identify the receptors that binds to IgA with Max Cooper in Alabama. So we've been challenging when I came back to France to understand how the receptors triggering the cell, either activating or inhibiting. But so the receptors, they have the motifs they call ITAM or ITIM, ITAM for immune receptor tyrosine-based activation motif, that has two tyrosines, you see in the top, XXL twice, that the motif that activate the cell, and the ITIM is half of that motif. So as you can see here. So we've been interested in the receptor that were isolated to the Fc gamma chain, is a homodimer that carries this ITAM. So as I told you, I was working in IgA, serum IgA in patients in health. And basically, since I start there, it was mainly involved in diseases like IgA nephropathy, AIDS, spondylitis arthropathies, hepatitis, several super diseases, purpura noxion line. And then it turns out that one of my students find out that a singular targeting of the receptor was doing a inhibitory effect. And then we looked at the literature, we found that anti-inflammatory effects of monomers, you know, IgA has particular to be monomers or dimers or trimers. You see on the right side, the polymers, the dimers or trimers, are most involved in activation of the immune system, whereas the monomer has been described since the 70s to inhibit the immune system. However, if you look at the receptors you isolate in Alabama, was a receptor that's associated with a homodimer that only can activate the cell. So what's how can IgA inhibit if you have an ITAM? That started with the antidogmatic role of the ITAM-bearing IgA receptor. And we have a collaboration with Magda Sampaio, Professor Magda Sampaio from the USP, for example, studying in pediatric cases and seeing that you have increased incidence of allergy and autoimmunity when you don't have the IgA, in IgA deficiency. So it is really seeing indeed what the people described in the 70s were really probably strong enough to see that indeed the monomers that you all care, the IgA in the serum, are playing an anti-inflammatory role. And the question that you have in our team is to say how this was working. And to make a very long story short, we find out that the ITAM that you see here is monophosphorylated, is partial phosphorylated when it's linked to the monomers. And when you have an antibody of IgA seeing an antigen, it aggregates and this activates strongly, as you see on the right side. When on the left side, you have the inhibition. So we have been able, in this paper that we published in 2005, to show really the monophosphorylation of the gamma chain, and that was really recruiting a very effective, good and very impressive effect at the phosphatase that causes SHP1, and this phosphatase is able to block heterologous receptor. So this monovalent ligand is able to really have this effect, explain the IgA monomer that is acting in different aspects. And when you aggregate the receptor, you stabilize kinases like Syk, for example, and this activates strongly the cell, as for other receptors, the TCR, the BCR, etc. So, for example, if you look at this paper that we published in 2015, you have several papers. I don't have time to go all over those papers. You see here, we did with Boehringer in Switzerland, they purified the IgA like they do for IV, IgG. And if you use this IgA very huge, very well purified, injecting animals that develop arthritis, after starting the arthritis, you see that really you can reduce the inflammation very strongly in the animals. You see in the right side arthritis plus monomers, IgAs on the right. And this is really confirmed that what we have done in vitro, in vivo, has been shown by arthritis, by glomerulonephritis, for asthma, all able to inhibit with IgA or antibody that targets the receptor monovalently. So how this was working inside the cell? That is very different from the team. You need to have, everyone has IgA sit to the receptor, and then it's in the haft, and then the activating receptor is not there. When you have signal 2, this is colocalized inside the cells, and you have the possibility for the kinases to be knocked down by the phosphatase. So you see here in blue, the SHP1, that is very close to the kinase for the heterological receptor that is knocked down by dephosphorylating the receptor. So this is also controlled by Src kinases, and you see that Src kinase lean is mostly involved in this we call inhibitor ITAM, and FIN is mostly involved in inactivated SHP1. So this is something that we published in 2017. Then it comes to immune evasion. So we then did several other receptors. In this case, Fc gamma 3 or CD16 is a receptor for IgG. And then we start to work on that, if this is true for other receptors. And then we, at that time, I have a Brazilian student that Irineu Velasco sent to us, sent the mice to the Faculdade de Medicina de São Paulo. And Fabiano had done several CLP. CLP is a technique to induce sepsis in animals. And very surprisingly, what he observed was the resistance to the sepsis was the animals don't have the Fc gamma, the gamma chain, this Fc gamma chain that carries the ITAM. So then you say, why? This is very unexpected, because you saw that it might be the inverse. So and then we try to understand the mechanism, and the mechanism, we look on the literature and find in 87 that E. coli was able to bind directly to the receptor, Fc gamma 3, CD16. And then I told Fabian, let's look for phagocytosis. And we see that indeed, when you don't have a gamma chain, you have almost 14 times more phagocytosis than the normal animals. And don't have a difference with or without opsonins when you don't have a gamma chain. Opsonins is the IgG anti-E. coli. So in normal individual, when you have antibody, you can induce phagocytosis. You don't have, the E. coli does not bind because you have a different immune system. So what we propose in this paper that we published in 2007 in Nature Medicine, that was E. coli in patients that don't have immune system, that are immune deficient. The E. coli can bind directly to the receptor and blocks phagocytosis as a mechanism to immune invade, to grow and to invade the host and to cause sepsis. And also blocks TNF, that's another very important cytokine. In people that has antibodies, obviously the antibody will be targeting the E. coli and disaggregated the receptors, and the receptor have be activated and do phagocytosis. And we don't, for example, the model, the animal, they don't have the gamma chain. You see the E. coli is binding to is not binding to, and then you have a stronger phagocytosis because you don't have any negative signal, so it's increasing. So this was an NR with the FAPESP that we have in 2012 with Irineu Velasco. And since that, Fabiano is working that in the Faculdade de Medicina de São Paulo and published a paper together with us, but mainly him, with his student in 2016. So in part 2 of my talk is mostly devoted to the bacteria from the guts, the commensals, basically the commensals that eat the mucus from the mucin, and how this can be involved in autoimmunity. And then we use that, the IgA nephropathy case. What is IgA nephropathy? And it is described by Professor Jean Berger, that is on the left. And he described that you have deposits of IgA in the glomeruli of the patients. And this is also associated with IgG in these deposits. These patients are very common. It's one of the most common nephritis worldwide, and very common in Asia. And is associated with bacterial infections of the upper tract, as hematuria, proteinuria don't have specific treatment. It's one of the first causes of renal failure. So I have a chance when I came to France to work with him, and we have a lot of biopsies, and we already checked the IgA deposit in the kidney, and the IgA was a different charge, electric charge, which we postulate in the discussion that was due to the glycosylation. It turns out by several other groups that indeed this patient has a galactose deficiency in the hinge of the IgA. You have the two arms. The hinge is an axilla here, a lot of carbohydrates. That's all linked carbohydrates. So in this area of the IgA, you lose galactose and sialic acid. And then it becomes an autoantigen. You have IgG anti-IgA, and this makes complex. We also been in my lab involved in receptors I showed before, the Fc-alpha receptor, and they're also called CD89, can be solubilized. We did that with Agnès in the past at Necker. And those immune complexes are very important to induce the disease. We now understand the whole of this receptor as well. And with a student that came from Brazil, Ivan Moura, we work on the transferrin receptor as a receptor that binds to those complexes in the kidney. So this is 4 hits of the disease. We have the galactose deficiency, the antibodies to the IgA that form immune complex, stimulate the immune system to activate other proteins like CD89 and also AIM, and then also drops to the kidney by receptors such as the transferrin receptor that binds transferrin but also binds IgA or CD89. So the question that we ask in the team is what is the origin of the Gd-IgA1? We know the familial cases are very rare, and the genetic heritance is less than 5%, and there's a lot of studies on that. And for example, one enzyme that is very important to link to glycosylation, when you have deficient, you don't have renal deposits. And we have one case now that we studied in Paris, and it's very interesting because a very, very huge amount of Gd-IgA1, but no disease, because the patient is born with this defect, so the immune system sees as a own protein. So you don't have IgG anti-Gd-IgA1, so you don't have the disease. So the question was, is gut microbiota in the origin of Gd-IgA1 in the 95% of the patients? So we did this with Rahul Sokol's lab, in my lab, in an cohort called microbigan, and we found that indeed we have dysbiosis that's different from patients that has uremia. You know that uremia changed the microbiota because you have a lot of toxic stuff. But if you compare groups of CKD that has uremia and those that have IgA nephropathy, you have a dysbiosis. And it turns out that one bacteria, so-called Akkermansia, I don't know if you can read it, the arrow here, was increased in this case. And we confirmed this by qPCR in the patients as compared to the other patients with other disease. And very interesting, we found that this enzyme from the bacteria is also highlighting in the metagenomic analysis shotgun. You see the phospho-beta-galactosidase. So then you look at the structure of the IgA. I mentioned to you this axillary stuff, a lot of sugars in the two arms. The core is very identical for the mucin. You know, the gut has a lot of mucus covering the surface of the mucosa, and those structures are very similar to from that we found the IgA. So you postulate that Akkermansia, that is a bacteria that eats the mucus to survive, was eating also those from IgA1. For that, we use, for example, the animal model that you have that develops spontaneous disease. We did that. One of animals was done with Agnès and I when we were in the Necker. And then we crossed those animals with a colleague that made IgA1 animals, and those animals developed spontaneous disease. When you treat these animals with antibiotics, you abolish the disease. So disease is spontaneous in the animal, as I show you. In this, you see the deposit in the bottom here. It's really very strong deposit on the mesangium, as Professor Berger has shown. This is CD89 and IgA1 deposit. And then if you treat with antibiotics, you abolish that. And if you now make a gavage with either E. coli, you don't have any disease coming back, but if you put Akkermansia in the monocolonization, you see the positive coming back, and also you see the albuminurides coming back. Antibiotic is able to treat the animals and to abolish even proteinuria. So we then we look on the patient to say what James Gleason has done. There's a student that just finished the thesis last year. He has treated the IgA, purified IgA with Akkermansia in culture. The Akkermansia was eating the sugar of the IgA1 to survive, and put in the Western blot and look with antibodies that recognize this deglycosylation form. You see here with Akkermansia, AKK is positive, E. coli is negative in the blood. And then it took serum from patients and put to the sheets of the nitrocellulose sheets for the Western blot. You see, only IgA serum is able to recognize the deglycosylate IgA1, but not serum of CKD or from health, confirming that what we discover in the dysbiosis and then applying to the animal model to prove the mechanism, and then coming back now to see the serum from the patients that is recognized IgA1 treated with the bacteria that was increased in the patient. So then the question is why it's increased, right? Why you have dysbiosis? Why you increasing Akkermansia and don't having explanation for that? And then you look at the GWAS that has several GWAS has been done in this disease. You see the two loci came, the defensins loci and card9. So defensins, we look and find out that alpha-6 defensin was controlling Akkermansia growth. And this was not known, and one of the reasons Science Translation Medicine took the paper, because really new information, very new information. And see, in non-IgA patients, CKD, you have, more you have the defensin, less you have Akkermansia. And in patients, you lose this correlation. And if you look in vitro, you can kill the bacteria with recombinant alpha defensin 6. And here on the right, and in bottom, with Pasteur Institute, we have been able to do electronic microscopy. So to finish my talk, just to summarize what's happening, if you look in the gut, so gut is controlling the disease, that's clear. If you treat with antibiotics, you abolish the disease. If you now put the Akkermansia back, you have an even stronger disease. In the submucosa, you have plasmocytes that produce dimeric IgA that transfer to the lumen, and then you have this mucin degrader that is enhanced by alpha defensin, decreased of the alpha defensin. We see if patient has a lost site with polymorphism, so have less alpha defensin, is not killed enough, and then is increasing alpha defensin, deglycosylating heterotranscytose back to the circulation, it becomes an autoantigen. And this autoantigen is the first time in the literature proving that indeed environmental factors can induce autoimmunity. Everyone thinks that autoimmunity is genetic involved, or postulation that virus also can be involved, but not formal proof. This is the formal proof that the bacteria can indeed be a cause of autoimmunity. So we recently applied a project together with people from UNIFESP and the USP and University of ABC. We especially with Professor Pacheco, Álvaro Pacheco, and Professor Câmara at the USP. And this collaboration with Nils Câmara has been for several years due to a student, Ivan Moura, that I mentioned for transferrin receptor, that unfortunately passed away in 2018. And we have been since 2012. So now the poll dropped to the INR and to FAPESP. This project, including mate cells, or Tania's presented yesterday, and this project involves Agnes as well, and the group from Harris Sokol. And you see in health individual, it has been published by a group that in Science Immunology that mate cells is involved in IgA production. So in card9, there is also our data that's in preparation can control like a break to the IgA production in the gut. So this is in health. Individuals with IgA nephropathy, you have defensin deficiency, so you have more Archaea invading the mucous area and activating tau receptors, and card9 is defective as well. So you have much more MATE. This is our preliminary data that increasing IgA. So you have not only deglycosylations, more IgA is produced by the MATE in the gut. And we postulate the bacterial infections that you have probably a lot here in São Paulo, that the cohorts in UNIFESP with Professor Pacheco will be studying the role of the MATE cells through those infected patients or not. So the cohorts now will be established if you have the grant. And the final to thank you is just to, before that, I will acknowledge my collaborators that is in this slide and the supporters, and thank you for your attention. I probably have a little bit over.
Thank you very much, Renato. Your talk was very inspiring, and everybody must think about prescribing antibiotics because of dysbiosis. That's very important. And now I'd like to invite Professor Amaro Neto. He is professor of the Department of Pathology. He's an ID physician and a pathologist. His research line is in pathology of infectious disease, autopsies, and this is caused by bacteria, virus, and fungal. Please, Amaro.
So, good morning, everybody. I'd like to thank you for inviting me to take part in this meeting. I'm going to talk about pathology of infectious disease in recent epidemics in the city of São Paulo, Brazil. And we are living in a world with a lot of One Health and global health issues, allowing the emergence of new infectious disease, emergent infectious disease, emerging infectious disease. And in the last 20 years since the end of the last century, we've been experiencing a lot of epidemics here in Brazil. Dengue fever is a long-term problem. Every season we have cases of dengue, and some in the last 20 years, some years with epidemic season. Nowadays, we are facing a severe epidemic of dengue in the city of São Paulo, the entire country, but you are here the most number of cases and even the deaths. H1N1, influenza, Zika, chikungunya, yellow fever, measles, COVID-19, syncytial respiratory virus, monkeypox, and again, dengue fever. And so, what's the importance of autopsy? Or what's the importance of anatomic pathology, pathology of infectious disease for epidemics? Autopsy is important to define cases, case definition, especially the severe cases that we can understand the virulence of pathogen. You can understand the pathogens of a disease. You can understand the distribution of an agent and all of the damage that can cause in all of the systems. To do the differential diagnosis, especially with disease that sometimes mimic some specific infectious disease in an epidemic. To understand the pathogens, to evaluate new therapies, including even vaccines as a pharmacodynamic vigilance, surveillance, and understand new therapies, for example, liver transplantation that I will show you something, and preparedness. All of this importance of the autopsy are, I think, it take part in the preparedness of entire society, entire health system to understand what happens with an epidemic. I can show to you in this slide many examples of outbreaks and epidemics that the first case, the index case, were discovered through the autopsy. For example, in AIDS, in the beginning of 80s in Los Angeles, New York City, many cases of young men with pneumocystosis, cytomegalovirosis, and 2 years after the beginning, they discovered the HIV that was the cause of the immunodeficiency. Tuberculosis in Ohio, 1990, the outbreak of leptospirosis in Nicaragua, the bioterrorism attack of anthrax in New York City in 2001, and one of the first cases of Zika virus described as an autopsy that allowed all of the researchers to understand the anatomical pathology, the diagnosis, the PCR, the sequence of the virus with the tissues collected during the autopsies. And for example, Zika virus would be discovered as early as possible here in Brazil if the cities that were affected by the virus, that the virus were circulating in the northeastern Brazil, if they had a system to understand, to study all of the stillborns with microcephaly in that area. And so the epidemic started by 2015, but would be discovered as early in 2013. And what about São Paulo? I work at the Hospital das Clínicas attached to the University of São Paulo. This is the biggest public hospital, public and university hospital in the Latin America. And the Hospital das Clínicas is a reference for treatment of severe infectious disease, not only severe infectious disease, but it's a tertiary, quaternary hospital. And in the last epidemics, we had a very important role to control, to treat, diagnose, to treat patients with yellow fever, COVID-19, measles, syncytial respiratory virus, and now dengue fever, severe cases that are referred to this big hospital. It's placed in the center of São Paulo. I think most of you know this hospital, and the visitors will take a moment in the hospital. In our pathology department, our role is to support the hospital in both surgical pathology and even the anatomical pathology and autopsy. We have the biggest morgue in the world. We perform more than 14,000 autopsies per year. By 500 cases came from the hospital, Hospital das Clínicas, but all of the other cases came from all of the hospitals in the city of São Paulo metropolitan area that do not have an autopsy facility. So we understand after 10 years of work in this morgue that the autopsy can be important to surveillance, to diagnosis, and to understand what happens with severe infectious disease. And we have many groups that we study at the autopsy department, but I am dedicated to infectious disease. We have groups dedicated to neoplasia, groups dedicated to cardiovascular pathology, but my specialty and my dedication is to infectious disease. And so I started talking a little bit about the yellow fever. We had an epidemic in 2018, 2019, 2 seasons. More than 500 cases, and with a mortality around 40% of the cases. We were the reference to treat those cases. And we start with the basic in the anatomical pathology, the macroscopy. It's very important to us to see the macroscopy and to understand that they have the first diagnosis, the first insights about the pathogens of infectious disease. At the liver, with steatosis, with this color orange, very congested after fixation, this pattern with a pale brown surface, cut surface, and at the microscopy level, you can see here the hepatocytes with steatotic degeneration and apoptosis. It's called the calcium rocha-lima bodies, with very few, very mild inflammatory reaction. And we published many papers about yellow fever. It's a research line. The first one was a comparison between minimally invasive autopsy and conventional autopsy. We studied the 21st cases and compared the minimally invasive autopsy with samples collected with a needle guided by ultrasound and compared with the conventional autopsy. And the results were the same. It was very important as nowadays I can see this very clear, but it was an important measures of preparedness in 2018 with this epidemic of yellow fever. Because after this, in the COVID-19, we were prohibited to perform conventional autopsy because we do not have NB3, NB4 autopsy rooms in the entire country. And so it was possible to us to study the case of COVID-19 with the autopsy with this method. So it was very important at that moment. And the next one important article that we published was to study 4 cases that were submitted to liver transplantation, the institution. I think during the yellow fever epidemic, about 15 cases were submitted to liver transplant. Many cases died. In our hospital, 6 cases were transplanted, 2 survived, and 4 died. You have the opportunity to study those cases, those corpses. And we saw an infection of the liver of the graft by the yellow fever virus. So that was a treatment, an urgent treatment, trying to save their lives, a very young man. And most of them died and did not understand what was going on in many institutions. And in our hospital, we did this conduct to perform autopsy to understand what was happening. And we saw the infection of the graft. Here, the same aspect that I showed you before, a liver with steatosis, ischemic necrosis, and at the microscopy level, the same mid-zonal hepatitis with apoptotic bodies and steatotic with a positive immunohistochemistry for antigens of yellow fever virus, and also PCR positive in not only in the liver but in many organs, showing that yellow fever is not only a pathotropic virus, but also a virus that can disseminate and infect many tissues. And as I said before, it's important for pharmacovigilance, for example, for vaccines. One of the main problems and main concerns about the yellow fever vaccination is attenuated virus, and can cause, in some cases, in 1 to 1 million cases, neurotropic disease, hepatotropic disease. And so the autopsy is important to define the visceral reaction to yellow fever virus. So this is a paper that we published at the Emerging Infectious Diseases, and we showed some cases with hepatitis, splenitis, here the spleen, meningitis, and myocarditis, nephritis, and hepatitis due to vaccinal strain of yellow fever. And now we are having a PhD student. The first one studied the heart and the yellow fever, 73 cases, vaccine strain, and also the Civatic strain. And we showed myocarditis, secondary infections due to bacteria and fungal infections, and also hemorrhage and edema in the conduction system of the heart, explain the Faggot sign that were described in the last century, when the patients with yellow fever and typhoid has bradycardia with fever. And so we understand that there is endothelial damage in the entire heart caused by the yellow fever here, stained with the immunohistochemistry. And this edema hemorrhages can impair the conduction in the conduction system, the electrical side in the conduction system. And now we are having an international partnership here with Dr. Adam Bailey from the Wisconsin University. We are establishing an experimental model of yellow fever. We have already one paper published about the coagulopathy, and now we are studying the effects of the yellow fever virus during the infection in the intestines and also in the liver. And after the COVID-19, as I said to you before, we performed during the 2 first years of the pandemics, minimally invasive autopsy, We performed more than 19 autopsies. It was possible to us to understand what was going on with patients with severe disease hospitalized at the Hospital das Clínicas. And we were very successful to be one of the first groups of pathologists to publish about the pathology of the disease, describe the diffuse alveolar damage, hemorrhages, epithelial hyperplasia and reactive, the presence of the antigens of the SARS-CoV in the epithelial endothelial cells, here stained in red by alkaline phosphatase immunohistochemistry reactions. And we showed thrombosis in the pulmonary vessels and also in our other organs, showing that COVID-19 was not only a respiratory infection disease, but it was also a vascular infectious disease with thrombogenic and vascular chronic effects. And here, monkeypox in 2022. Brazil, again, was one of the countries in the world with the outnumber of cases and also death. We ranked as the second country in the world with the largest number of cases and deaths, especially those patients with AIDS with low TCD4 count cells. And one of the main questions during the monkeypox epidemics in UK, Europe, United States was how those patients are dying. What's going with those patients? They are dying due to another opportunistic infectious disease associated with AIDS, or they are dying due to infection of the monkeypox. As the autopsy rate in the entire world has declined along the last 40 years, it's not used in the United States and even in Europe to perform a large number of autopsies and to be ready to perform autopsy in all of the cases. So it's a medical procedure that's rare. Many families do not accept to do the autopsies, and there are a lot of issues to pay the autopsy, the procedure. And we had the opportunity in 2022 to examine 2 cases of AIDS of young men. Both of them, they were not aware about their HIV status. They had very, very low TCD4 count, less than 20 cells per microliter. And they were hospitalized at the Instituto Emílio Ribas, that is also placed in the center of São Paulo, near to our hospital, in the Dr. Arnaldo Avenue. We examined 2 cases and we observed many skin lesions, here typical lesions of monkeypox, but in those cases with AIDS, and this observation was also seen in UK, United States, but they were not described what was going on in the pathological level in microscopy. And examine the cases here as the back of a patient, one of the patient with HIV and 10 cells, TCD4 cells. And they were developing during his hospitalization progressive lesions that were confluent, very necrotic with this aspect of target, encompassing more and more lesions with a centrifugal pattern of the spreading. And here, the microscopy, necrotic cells, the borders with typical aspect of monkeypox lesions, and we found a lot of antigens of the virus affect not only the epithelial cells, but also mesenchyma and vascular cells. And we described a systemic vasculitis provoked by the monkeypox. Here, the tongue, the larynx, it was published in this comment in the Lancet Infectious Diseases. Gastritis, esophagitis with similar cell lesions as you can observe in the skin. Colitis, here another case that another case that was submitted to colostomy. Here, the rectal region with proctites, with infection of all of the tissues around the rectum, and infections of epithelial cells, mesenchymal cells, the nerves, and the vessels with vasculitis. So the virus is ubiquitous in this kind of patient with a very low TCD4 cells. It's very important to contain the spread of the virus. And the behavior is in monkeypox disease, in HIV with low TCD4 cells is an opportunistic disease. Here, pneumonia with necrotic pneumonia, nodules of necrosis. And so I'm getting to the end of my presentation. What's the next epidemics here in São Paulo? Maybe Maiaro. There is a long term of prevision that Maiaro could reach our region. It's an endemic viral disease in the Amazonic forest. Could be West Nile. Could be an important case of travel medicine. If depends on the point of view, but we can see influenza H1N1, COVID-19, Mpox as a traveler medicine problem. And you can see a rise of cases of endemic disease, for example, spotted fever in the 2023, here in the region of the São Paulo, Oropush that reached our regions, Rio de Janeiro and Vitória Espírito Santo. And endemic diseases such as tuberculosis, porotrichosis, leishmaniasis that are a huge problem in our region, zoonotic disease, mycotic or opportunistic disease, for example, aspergillose, mucormycosis during the COVID-19, and also dengue. Since the beginning of January, before more than 220 autopsies of dengue disease in the capital. We are trying to finish all of the cases to write the first report and submit the results of those autopsies. We are working together with Professor Ester Sabino and the infectious disease department, but we can see also a target for Sudan virus, H5N1, Nipah virus that emerged infectious disease in India, and the failure of early detection of infectious disease in the field of anatomical pathology can be due to little knowledge about the histopathology to recognize the pattern and give the diagnosis, and no available methods to in situ diagnosis. This is what I'm dedicated to do. So we launched in 2022 a discipline, the post-graduation department program. We are studying this emergent and re-emergent infectious disease, even those that we already do not have here in Brazil. And this project that I would like to share with you, Prepare, in English would be Preparedness in Anatomical Pathology for Rapid Response in Epidemics. In Portuguese, it's the same. Preparatório em Patologia para Resposta Rápida em Epidemias. We consider that our morgue is very, very important, more than 14,000 autopsies. We were the epicenter of many epidemics here in the Brazil, and so we have to be ready to give this diagnosis as soon as possible to communicate the epidemiologic authority, health authority, and conclude the autopsy and establish a network between labs, anatomical pathology labs, able to give those diagnoses. So we got a funding from the CNPq, a federal funding, and we are following the blueprint priority list from WHO. That was, there is a new listing that was published in the end of 2022. COVID-19, we have cases, H5N1, we do not have cases, Crimea, Congo, Ebola, Lassa, Nipah, Rift Valley, Zika, and the disease X, that could be any disease. And also we are dedicated to severe infections that are endemic here in Brazil. It sometimes can cause epidemics. So our focus is the in situ diagnosis. We start with the microscopy, as I showed you. hematoxylinase in the basic stain in anatomic pathology, the special stains as Grocott and other stains to detect mycobacteria, as UNIFESP, for example. Immunohistochemistry is something important to us because all of the labs, anatomic pathology labs in the country, they are able to do the immunohistochemistry. And of course, hybridization, electron microscopy, molecular tests such as PCR. They are not available to all the labs, but we are focusing in those methods. You can also do virus sequencing, bacterial sequencing in our labs with the tissues that you can get during the autopsy, transcriptome. And so we are working together with other groups, not only the anatomical pathology. And this is the idea, to be prepared to give rapid diagnosis and understand the physiopathogens of infectious disease that you still do not have already, but it can be possible to get here. Thank you.
Thank you very much, Amaro. Everybody knows that there is no monotony in infectious disease life because it's very, very dynamic. And Amaro, thank you very much for our presentation. Now I'd like to invite Imane Meouche. I hope to pronounce your name correctly. She's a researcher from INSERM, and she's going to talk about the in vitro and in vivo approaches to study antibiotic tolerance in E. coli.
So thank you so much for the invitation, for the opportunity to talk here, and also for the introduction. So I'm Imane, and I will first tell you a little bit about who we are. So I'm an INSERM researcher in the same building, actually, than the Renato Center. And we are the YAM unit. We're located in the Faculty of Medicine in Bichat, near the hospital Bichat. And YAM stands for Infection Antimicrobials Modeling and Evolution. And it's really an alliance between medical and basic research. We're a unit of 4 teams. We're interested in very interdisciplinary research, going from bacteriology, virology, prevention, epidemiology, evolution, modeling, biostatistics. So if you're really interested, you're really welcome to visit the website of the unit. And I am in the Everest team up there. So we're interested in the ecology evolution of virulence and therapeutic in Escherichia coli. We're not only interested in Escherichia coli, there's already actually some collaboration between researchers in Brazil and researchers in our team. So for my group, we're really interested in antibiotic resistance, which all of you know it's a major health problem. And here, as you can see in this graph, bloodstream infections that are often a complication of urinary tract infections are increasing over the years. And also the problem is the number of isolates that are resistant to antibiotics is also increasing. So antibiotic resistance is a major problem. And it's often due to genetic modifications. So when we think about antibiotic resistance, we think about the acquisition of resistance genes and mutations in the chromosome, etc. But antibiotic resistance is not only due to genetic changes, it can also be linked to phenotypic changes, a phenotypic heterogeneity. So as I'm showing you here, a population of bacteria. So you all imagine that if this population have the same DNA, the same genetic material, it will be identical, but actually no, because there is some phenotypic heterogeneity that exists between cells, even if they do have the same DNA. And this phenotypic variability can be due to the environment, to partitioning of molecules between daughter cells, and also can be due to noise in gene expression and the stochastic nature of transcription and translation, etc. And during my postdoc, we were really interested in this phenotypic heterogeneity between single E. coli cells and how this affects survival of the population. And here I'm showing you a video, it goes fast, sorry, but a video of E. coli cells grown in a microfluidic chamber. And we add antibiotics to kill the cells. And when we add antibiotics, you will see in red, we don't kill the cells at the same rate and at the same speed. So the cells, even if they do have the same DNA, they're not killed actually at the same speed. And when a filament stays at the end, we can see that it does grow back again and divide even when the antibiotic is removed. So this is a problem in terms of relapsing infection, etc. So during my postdoc, we were really interested in this phenomenon, and especially the genetic causes actually. And we were interested in MAR-A, which is a multiple antibiotic resistance activator. Why are we interested in regulators? Because they're expressed at a low level in the cell. So their heterogeneous concentrations or quantities in the different cells is really important. And they also affect the expression of a lot of downstream genes. For example, MAR-A regulates the expression of a lot of resistance genes, stress resistance genes, for example, efflux pumps that are very important to export antibiotics and other compounds from the bacterial cell. So during my postdoc, we studied the variability in MAR-A expression between isogenic cells. So we did a lot of microscopy and showed that MAR-A expression is variable between single cells growing. And this expression was also correlated to survival. But some people can say, okay, it's transient resistance, but cells will die at the end. But actually, we also showed in another study that this phenotypic heterogeneity was linked to more permanent changes. So just in brief, we showed that the cells that expressed more MAR-A or more efflux pump at the same time expressed less MUTS, which is an enzyme involved in mismatch repair. So this transient and phenotypic variability and transient resistance can also be linked to more permanent changes and actually in the emergence of permanent antibiotic resistance. And when I came back, I actually wanted to study another facet of antibiotic response. So in the absence of antibiotic resistance, many bacterial infections still relapse. So we, like the susceptibility test measurements actually to know if a population of bacteria is sensitive or resistant to antibiotics is doing antibiotic standard antibiogram, sensitivity test, etc. And we know actually that even if bacteria are sensitive to antibiotics, we sometimes have treatment failure and we sometimes have infection relapses. And this is really particular, for example, in the case of urinary tract infections. Urinary tract infections are very frequent. One woman over two has one infection during the year, and they relapse at a high frequency. And UTIs are mainly caused by Escherichia coli, which is a commensal but also a major agent causing UTIs, which makes it as well a major health problem. And the reasons of relapses are actually multifactorial, right? So if a surviving bacterial population is here, it can be due to different factors. It can be due to the immunity, and you heard a lot of talks on that. It can be due to pharmacokinetics of the antibiotics, where the antibiotic can reach or not the site of the infection. It can be due to environmental factors like pH, the iron concentration, etc., that are in the site of infection. And they can also be linked to the population dynamics and the bacterial response. And this is what we are really interested in terms of growth rate of the bacteria and the inoculum size, also of the physiological state of bacterial cells during the infection. So we're really interested in tolerance and persistence of Escherichia coli. So as opposite of antibiotic resistance, where bacteria can grow in presence of inhibitory concentrations of antibiotic, they just keep growing. Antibiotic tolerance and persistence occur when bacteria survive in presence of very high concentrations of antibiotic. They don't grow in presence of antibiotic. They're just here, sitting and waiting until the antibiotic is removed. And they only grow when the antibiotic is removed. So they are transient phenomena that are not very clear in susceptibility tests. When we perform MIC tests to calculate the inhibitory concentration, minimal inhibitory concentration, you can see that susceptible, tolerant, and persistent bacteria do have the same MIC, unlike resistance. But actually, when we go further and perform, for example, time-killing curve, we can see that a tolerant population is killed way slower than a sensitive one. So we spend more time killing 99% of the bacteria. And persistence is just a drug, it's just linked to a subpopulation, a very tiny fraction of the population that can survive for extended period of times during the antibiotic treatment. So we're really interested in this phenomenon and their implication and their relevance in UTI, if they contribute or not to relapsing UTIs. But before studying their mechanisms, we actually wanted to know if they are relevant, as I was telling you. And tolerance and persistence are linked really to the metabolic state of the cell and also to growth. For example, there are a lot of antibiotics that only target dividing cells. If the cell is not dividing or dormant or in a quiescent mode, it's not gonna be killed by antibiotics. So before studying the mechanisms of tolerance and persistence, we actually wanted to know how E. coli is behaving in vivo during a UTI. And growth and cell division are really hard to measure in vivo, right? In mice models, for example. So we chose to apply a synthetic biology approach that was developed in the US, where I'm not gonna go into the details of the genetic circuit, but in brief, it's a genetic circuit composed of a trigger and a memory element. And when we add an inducer, if the bacteria are dividing, they will express LAG-Z, so beta-galactosidase, and we will have blue colonies on plates. If they are not dividing at the time of the exposure, we will have white colonies. So for us, it was really a nice way to see if the bacteria are dividing or not during an infection. So what we did is we infected mice. We have a pyelonephritis mice model where we infect mice with E. coli. We have an infection in the bladder, we have bacteria in the urine, and we have an infection in the kidneys as well. So what we did is we actually calculated the bacterial load in the kidneys, in the urine, and in the bladder. And in blue, you can see the actively dividing fraction in the different sites of infection. So you can see here over time that in the kidneys and the urine, the majority of bacteria are actively dividing. And here we just did it with 3 isolates, so we can actually generalize a little bit the phenomenon to different clinical isolates involved in UTIs. But what was interesting also for us is in the bladder, after day 1 of infection, the bacteria stops dividing, and a majority of bacterial cells are in a quiescent mode, forming reservoirs, and this was also observed in the 3 strains we tested. Which was really interesting for us is during one single infection, bacteria are really doing different things in 3 different microenvironments. And we wanted to know if this heterogeneity in bacterial division correlated with heterogeneity in survival as well. So what we did is we infected mice with E. coli, and then we treated them with antibiotics. We treated them with cefotaxime, which is a beta-lactamine that actually acts on dividing cells. And we can see that in the kidneys and in the urine, we can significantly eradicate bacterial cells, but in the bladder, we could not eradicate bacterial cells. We said, okay, let's use phosphomycin, which is widely used in cystitis, and we exactly observed the same thing. We can kill bacteria in the kidneys, in the urine, but not at all in the bladder. And then we said, let's use quinolone, which maintains some activity on non-dividing cells. Then we actually observed the same thing. In the kidneys and in the urine, we're able to eradicate bacteria. In the bladder, we are not able to eradicate bacteria. And we said, okay, are the bacteria intracellular in acidic pH? And ciprofloxacin doesn't work well in acidic pH, so let's use another quinolone that works better in acidic pH. We used delafloxacin, and it's actually the same thing. We could not eradicate bacterial reservoirs in the bladder. So what we're doing now is where the bacteria are, and which was really interesting for us is that every cell that survived, or most of the cells that survived, were not dividing. And also, they all have the same MYC. So we didn't observe any emergence of resistance, even if we do multiple cycles of antibiotics that we also tried. And also, we did whole genome sequencing. We didn't really see any mutations, even in tolerance or persistence genes. So what we really can conclude is the phenotypic, the phenotype of bacteria during the infection and the treatment are really important as well and need to be taken into account. And also the interplay between bacterial cell divisions, antibiotic response, and and the environment. And what we said, okay, let's take the problem in a different way. And we have a collection of clinical isolates in the lab. We measure their tolerance and persistence phenotypes, and we took one isolate here in green that actually had the same MIC than a sensitive strain, but does survive more if we perform time kill curves. So it's more persistent and tolerant to antibiotics as well. And we infected mice with this strain. And while we observe that in the bladder, it's independent of the strain we use, in the kidneys, we actually found a relevance of this strain that has a high persistence level. So persistence in our case was really important in the kidney, actually, where we here cannot significantly kill bacteria. So we are in the process of studying the mechanisms, the genetic mechanisms of tolerance and persistence. And tolerance and persistence are really widely studied. They're less studied than antibiotic resistance, but they're mostly studied in K12. And K12 is a E. coli lab adapted strain, and it's very good, it's very known and studied, but it doesn't represent the whole genetic diversity or even the phylogenetic diversity of E. coli. So for example, K12 has around 4,000 genes, but the natural isolates, we have between 4,000 and 5,000 genes. So we can, even more than 5,000 genes. So K12 doesn't represent this whole diversity of the clinical isolates. And we are looking into determining the mechanisms of adaptation and antibiotic response in clinical isolates now. And also we are interested in different environments. For example, we're studying bacterial response in natural media like urine, which we believe it's more relevant as well to UTIs, and also different carbon sources, different environment as well. So it can tell us a little bit about the more mechanistically the antibiotic response in bacteria or in E. coli here. So what we have in the lab, we have a large collection of clinical isolates, of natural isolates, with low subcultures. So we don't have adaptation to the lab environment actually. We maintain genetic diversity of the strains. And we perform time kill experiments, as an example here, on ampicillin-sensitive strains. And we can see, even if the strains are all sensitive to ampicillin, we can see a very heterogeneous response to antibiotics, to ampicillin. If we add high concentration of ampicillin, we don't kill the strains similarly, actually, even if they are all sensitive. So what we are doing now, we are doing transposon sequencing to study the mechanisms of tolerance and persistence in vitro and also in vivo on the different set of clinical isolates to see if the mechanisms of tolerance and persistence are shared between isolates or if they are very different. We are also doing genome-wide association studies on larger strain isolates to also study the genetic determinants of tolerance and persistence. And of course, we will test that in our UTI model to always parallel the in vitro and in vivo. And we are also going further to strains that we collect from actual patients who had recurrences, recurrent UTI actually. And we are studying the microbiological and sometimes the patient, if they have some also host factors involved. But we are mainly focused in the microbiological factors that can predispose or help strains or cells to persist in presence of antibiotic treatment and cause relapsing infections. So finally, I would like to thank all the people involved in these different projects and the team. And please, you can contact me if you have any questions or ideas, and also look at the website and contact us for further collaborations. Thank you.
We have 10 more minutes, so I would like to invite Renato, Amaro, and Ester to join Imane at the table. And if we are open for questions. Thank you very much for the presentation, and it was wonderful. I like a lot.
I'm not sure. Does it work? It doesn't seem to work. Okay. Yes, so I would like to have a questions for Imane, because in fact, with the same questions. Do you have some idea why the bacteria doesn't grow or that divide in the bladder as compared to urine? And then the other thing is, in fact, in real life, when the women are infected, they get antibiotics, they can recover. Donc that means they can kill probably bacteria in the bladder, or it's only immune-derived, that the immune system can get removed the bacteria in the bladder without the help of antibiotic, which act rather in the other tissues.
Thank you for the question. So for the first one, for the
No, it's on. Just talk too close.
So yeah, so for the first question, we think the mechanisms are not really known, but some studies started to show that bacteria are exposed to reactive oxygen species, reactive nitrogen species, that makes them like also dormant. For the second question, yeah, when we treat, we can actually decrease the bacterial load, and some women actually do. They're fine, but we know that bacterial reservoirs can stay sometimes for months and years. And for like the parallel, what we know, it's because it's hard to do biopsies actually. So we know in the urine that from the urine that we know that some immune cells or epithelial cells have bacteria in them, and for a long period of time. But it's actually, we don't know if and when the second episode can happen, and it's a mix, I think, of bacteria dividing again and infecting, and the immune response as well.
Hi, thanks for the brilliant presentation. My question is for Renato. So I wonder whether you could develop a bit more about the role of Akkermansia on the IgA alteration, and more specifically on where you think the bacteria is active. So it's the bacteria that invaded the mucosa or in the lumen?
Well, yes, a good question. Actually, look, we didn't have time to present all the data, but in the mice, the bacterial dysbiosis occur in the ileum and the colon, that you have more Akkermansia, but you also have other species, Victivalis, also Huminococci, that all those species need to eat the O-glycans from MUC1 and MUC2. So that is, we believe in the patients is due to the absence of the defensin, but it may not be the only cause for that. Can be other causes that might be environment mediated, which is, I don't know if you answer a question precisely, yeah? Yeah, partially, yes, okay.
Yeah, we can talk a little.
Now what we need to do is to look in patients. But with IgA nephropathy patients, to get colonoscopy biopsy is a challenge what James is doing with a group of pathologists in UK, in England, in the United States and France, and Ireland. He's Irish guy. And we are trying to do a spatial analysis of the DNA in the biopsies of the colonoscopy of patients that go for routine colonoscopy. We plan to do with Mateus, but we couldn't do it here. But I think that can be done here also as well. We may do this with Álvaro at Hospital São Paulo. We are planning to do that actually also.
Thanks.
Congratulations for the outstanding
Presentations,
outstanding presentations. I have two comments in question. First, I think that it is a very good example why we should have infrastructure of research and how this was extremely important during this pandemic. I think that your work, Esther, was outstanding, and I think that Brazil deserves a big thank to you and your group for all the work that you have done. And the second thing is what Amaro is working. I think that Amaro, for me, that as a physician, it seems that you are a person of extinction in terms of future, because pathologists are, we are doing less and less autopsies, And this is, I think that you demonstrated how important it is to have people prepared to do that. So, and I think that your initiative to work as a team and to detect rapidly is outstanding. And I have a question, and in fact, this is, I'm not in the field of DI, ID, but I have a mom that has many urine tract infections, and the nephrologist, when I try to cure the symptoms of my mom, sometimes they say that if you have bacteriuria, you don't have to treat every time. So this is very interesting finding that you have tolerant, dormant, or whatever bacteria in the bladder. So these, is there any relationship with this type of bacterial types and the occurrence of symptoms? So these dormant are the ones that are kept there, and sometimes you don't have to use antibiotics, or just a stupid speculation?
No, I think, yeah, I think, I mean, as we saw from using antibiotics, they didn't help actually to reduce the bacterial load and to cure this infection. And we had an experiment as well where we did multiple cycles of ciprofloxacin, and even despite 4 cycles of ciprofloxacin treatment, we couldn't treat these reservoirs actually. So I don't know how much antibiotic will help in clearing actually the reservoirs if we don't have an extreme high like bacterial load in the urine or even in the kidney in the case of pyelonephritis. For the strains with bacteria, there are some research that is showing now kind of a competition between some strains that are in the urine actually, where there's bacteriuria, and actual UTIs really with more pathogenic strains, where they can compete. The strains that are usually sometimes with asymptomatic bacteria, for example, they sometimes are not able to invade well the bladder epithelium because they don't have sometimes the virulence factors that the strains from the phylogroup group B2 can have, etc. So I do think it depends really on the strains, and on the strain, and it's hard to say if we should just like keep it like this and not treat with antibiotic, because they will not treat it anyway. But I don't think that the antibiotic is able to actually clear reservoirs, unfortunately. That's why we're studying on when they are researching to treat them, for example, or even trying to understand the immunity behind that. So how can the immune system help the antibiotics as well? Or people are studying some phage therapy, some molecules that can penetrate in the urothelium, because we show also, and present everything, but we show a very high permeability barrier in the bladder, where sometimes when we add Triton to exfoliate bacterial cells, we can kill them better. We still have dormant bacteria, so there's a part linked to the physiology of the cell, but there's a big part linked as well to the urothelium barrier.
Hi. Thank you. Thank you for the great presentation. Very nice panel. I have a very quick question for Professor Amaro. You showed some very interesting results about ultrasound of autopsy and the association. That is quite challenging doesn't matter what kind of diseases we are tackling to see the association. So how do you do these co-registrations when you have these samples, ultrasounds, and I suppose you're comparing with the tissue? How do you do that?
Could you repeat the question?
The question was about the ultrasound samples which you showed, like the samples of autopsy, having the ultrasound, and you're trying to study the association.
The comparison between
Yes.
the ultrasound, the ultrasound and the conventional autopsy.
Exactly. Yes.
Okay,
So how do you do this co-registration? Because it's very challenging.
Yes, we at that time, 2018, during the yellow fever epidemic, we were established the minimally invasive autopsy, and so we compared this method with the conventional method. and so we
Good correlation.
start getting the written consent from the family, and the patients go to a room with ultrasound and tomography, and we get the samples with the needle. And after the minimally invasive autopsy, the body, the corpse goes to another room, and we perform the conventional autopsy. And all of the pathology that the pathologists that perform the one procedure do not talk with the other conventional. They gave the diagnosis separately, and so we make the comparison. At that time, we did a comparison; it was almost the same. And but in the COVID-19, we did only the minimally invasive autopsy. I answered your question. Yes.
Thank you very much for the audience. I'm just going to close the table, but before closing the session, I would like to say that's very important, as Esther showed, like preparedness, to be prepared. When COVID happened, Esther has already prepared the team to do the procedure. So as João pointed out, it's very important to support research and to prepare the team, because when the outbreaks occurs, everybody will be prepared to respond. So if there is no investment in the team, in the support in research, there is no preparedness. So I think that's very important. Amaro, I think that your work is amazing. I have seen the work that Marisa and the group has performed, and they have a magnetic resonance that they use for autopsy in HC. They have been doing an amazing work, and I think that's very, very important, the minimal invasive autopsy, because nowadays most families, they don't allow us to perform an autopsy, and it would be very nice because we could collect the samples all over the country and send them to Amaro, and Amaro or the group could perform this test. So I think that would be very important. And Renato, I think that maybe the manipulation of the microbiota, or maybe you'll be the answer for not only infectious disease, not only for the treatment of Clostridium difficile infections, but also for non-infectious disease. So I was very interested about the Akkermansia story. I think that was very nice, but maybe the manipulation of the microbiota will be the answer for many cure or at least control of infectious disease. And Imane, I think that your work is amazing. I have some suggestions. I would like to have asked you about the internalization of the bacteria in the cells, and if you have performed transcriptomic, and if you have used a different antibiotic that I'm gonna to ask you during the cough break. I'd like to close the session, invite everybody to have the coffee break on the second floor. Thank you very much. It was a very nice session.
Thank you very much.
A big applause for the speakers, because the speakers were very good. So thank you very much, guys.
Thank you very much.
Good morning, everyone. So let's start the neuroscience session. And I would like to thank FAPESP for participating in this very important event. And I would also like to thank Leandro Colli, who asked me to give the first talk in this session, as one of our colleagues from Paris could not make it. At least this unfortunate event allowed me to present a new topic in this very productive, productive event, and I will look forward to helping to strengthen the collaboration between Brazil and France, especially in the field of neuroscience. So I'm also from Ribeirão Preto, and you have already seen our bright and vigorous forest around our campus. So we believe that besides research, we also are deeply committed to combat global warming. Okay, so I will talk about some physiological signatures in depression in resilience, but also I would like to start with talking about our epilepsy surgery center. We have a very active epilepsy surgery center in Ribeirão Preto for drug-resistant patients with epilepsy, and we perform practically all state-of-the-art procedures in this area, as well as clinical pathology studies. So among these studies, perhaps a set of studies that we dedicate a large part of our time is temporal lobe epilepsy, which is the most common type of drug-resistant focal epilepsy in adults, especially in developing countries. So just a brief overview from this condition. Usually the patients during the first years of life, they have an initial precipitating injury, mainly febrile seizures, but could also be other conditions that affect brain, and through excitotoxic damage, we have damage to the mesial structures. So by the end of first decade of life, or beginning of the second decade of life, they can recover from this event, but they can recover, and they cannot have seizure, of course, but a large proportion of these patients develop what we call complex partial seizures, or now we call perceptive seizures. So usually we treat them with anti-seizure drugs. We can have control of them, but many of them develop pharmacoresistance. And so we go with these patients, and when we have certain that we are targeting the lesion, we could do a resection, and we have most part of the mesial structures. And this piece of tissue offers a great opportunity to study the underlying substrates of epilepsy and the associated comorbidities. Yeah. In yesterday's presentations, I was really impressed with this cutting-edge molecular techniques that people from the immunology and cancer field are using. And I hope that during this next coffee breaks and lunch, we can discuss, because we are still using more or less what Amaro has been using in his studies in pathology. So we have been involved in several studies, and sometimes as a neurologist, a clinical neurologist, I dare to work as a neuropathologist, and we have carried out several studies addressing the histopathological substrates that would correlate with changes in imaging and regarding especially the post-operative outcome and also the psychiatric comorbidity. So we use studies that we could correlate and have histological and imaging and see which are the structures, matrix molecules, or even markers of synaptic reorganization that we can also explain changes that are occurring in the neuropilon or in the matrix of these areas. And also looking at some things that are, in some of these patients, they have memory preservation, and we look at these subgroups of patients that, despite having damage in the hippocampus, they can have preserved memory, or at least less damage. But another comorbidity that we have been looking for in among the psychiatric comorbidities, depression is by far the most common subgroup of these symptoms, and it could could occur in approximately half of these patients, especially in temporal lobe patients. So we also have done studies with some neurotrophins and other markers, and doing correlational studies, establishing correlations between these markers, neurotrophins, inflammatory markers, stress-related proteins, and markers of synaptic reorganization in these various comorbidities. But I think that although it's very interesting, these studies are correlational, and they do not allow for advancement in understanding mechanisms. So that's the reason that we need translational studies. So I'm going to talk about depression. And the main conception present in literature and also in society in general about the etiology of the depressive symptoms was formulated as a hoc hypothesis based on the mechanisms of especially antidepressants. So they use that as the drugs that inhibit the recaptation of serotonin. But it was also previously proposed that would be based in alteration of these levels that would be associated with before noradrenaline and now with serotonin. And also, this is a general concept, that because who in the audience has never claimed that you need to eat a chocolate bar to be happy? So this is a general concept. But as we see in this Arvid Carson, this a Nobel laureate in 2000, together with Eric Kandel, in his lecture, he mentioned, however, it must be recognized that the brain is not a chemical factory, but an extremely complicated survival machine. So an alternative hypothesis for this chemical imbalance, it would be the neural network hypothesis, which claims that it would be the circuits or some circuits, especially structures in the prefrontal cortex, amygdala, and other accumbens and other structures that would be involved in this circuitry. And it allows also to understand better and maybe use other approaches that just using pharmacological drugs that would act in some circuits that would be the interested, but also we usually find several side effects because it's acting in the whole brain. So then I will just talk about the study that we used is the triadic model developed by Martin Seligman and Stephen Meyer in the late 60s. And it was also known as the theory of learned helplessness. And it was initially developed in dogs from the psychology experiments. They studied the responses to stress and the control or lack of control of these animals in adverse situations. So the paradigm is very simple, and we have three situations. The control situation, where the animal is not subjected to any shock. So in a situation that the animals would be submitted to the same shock, but with the difference that only one has control for this stimulus. Then 24 hours after that, the animal which has control for that situation, in a second situation, he is able to jump the fence and escape from the electric shock. And the animal that does not have control, have control, he assumes a helpless reaction and become desolate and does not act as an escape behavior. So this is what Martin and Seligman named learned helplessness. So the animal, another interesting thing in this, that the animal is helplessness. But if the same animal, if you experience before this paradigm a controllable escape, and then 24 hours after that, the animal is subjected to the stressor, then at the next situation, he was able to escape. So this is what we call behavioral immunization or learned resistance. So it is important to mention that both Meyer and Seligman felt very distressed at the beginning to work with dogs. And both considering, they wrote in very articles that they were dog lovers. And very soon, they also replaced all these studies to rodents, and several studies using this paradigm to have these three conditions and see for some psychological factors that could induce changes in this animal behavior. For example, animals with uncontrollable stress, they would present a median size of gastric ulcers more than control and escapable and controllable stress animals. So then we have this situation that we have in this triadic model. We can address the conditions that we have controllable stress or uncontrollable stress, and all the changes that is a consequence of this. Like, as I said, ulcers, or fear and anxiety, apathy and subordination, drug abuse, reduction of neuroplasticity, and also the occurrence of tumors. And this is a link between these stress situations and the inflammation response. And in opposition of this, you have the controllable. And probably you have heard about this comment in this paper that was published last month in Nature by the Charles Zucker's group in Columbia University, and was really breakthrough because they were they discovered that there are some cells in the brainstem, especially in the vagus nerve, that act as a master regulators of the body and in inflammatory response. So I think that news is not here, but yesterday he said that the inflammation group is the most important thing. But I would say that I knew that central nervous system would control those tricky cytokines and lymphokines that were presented yesterday. And as I mentioned to Anna, and she said, it is almost Greek to me. It also was almost Greek to me as well. So, but I think that this is an urgent call to talk about neuroscience in immunology. We have last month a paper in Nature connecting these two situations, and I think it's a great opportunity to talk about this interdisciplinary research. So back to its triadic model. 50 years later, the authors reviewed this model and commenting what was the progress in these 50 years. So originally, they have this original hypothesis that the lack of control is learned, and that would lead to decrease of escape, increase of fear and anxiety and drug reward, and other changes. But now we know that we have many structures that respond to stressors, especially in the brainstem, like a bandula, locus coeruleus, and other structures. And the dorsal raphe nucleus is the main structure, that is a serotonergic structure that would integrate and act as this target to these behaviors. But now we have this the identification of the prefrontal cortex as a control structure that controls dorsal raphe nucleus, and this the revised hypothesis says that controls is encoded by a higher level, and the helpless is the consequence of the stress in the absence of control. So this was a very elegant studies that this Myers and Seligman's group that did pharmacological studies modulating the prefrontal cortex, like, for example, inhibiting the prefrontal cortex in resistant animals, and they became helplessness. If animals with helplessness behavior, if they activate the prefrontal cortex, they become resistant. And also with this behavioral immunization, if we could inhibit this prefrontal cortex, the animals that were resistant became helplessness. So the idea of our group is just to see this, how stress or changes would lead to depression or resilience. And of course, in humans, this would be a maladaptive appraisal or an adaptative appraisal. And so, and this would be a consequence of circuits. So, and then in this graphic, we know that when you have control, positive balance, and safety, you would have a positive appraisal, so we would be more resilient. On the other hand, if you have uncontrollable situations, negative balance, and you are at the risk, you are not safe, you would lead to a situation that would lead to depression. So we are going to talk about contrability, and working is in this model. So just a quick review to phylogenetic changes or differences between animals and humans. So we have stressors working in dorsal raphe nucleus, projecting to these prosencephalic structures. And in the control situation, we have this dorsal rapid nucleus in the humans and animals, and we have the prefrontal cortex is a huge difference, but we have these structures projecting to dorsal rapid nucleus and other structures like the hippocampus, inhibiting and decreasing this helplessness behavior. So the question that we have is this: do we have electrographic or electrophysiological signatures that could describe these animals? So the goals of this project was to characterize differential and controllable and uncontrollable stress, and see if electrophysiological activity between prefrontal cortex and the CA1 region of the hippocampus could identify these patterns. So that's we have a laboratory of electrophysiology, and we do studies in optogenetics, but also in local field recordings and unit recordings. And we, with this approach, we were trying to cracking the neural code of resilience and see if using this paradigm of the triadic model, we could identify these changes. So we use all the approaches that machine learning allowed us to either unsupervised or supervised techniques to discriminate changes in these EEG patterns of these animals, like factorial analysis, hierarchical clusters, linear discriminant models, artificial network, and see if we can discriminate these two situations. So what we did is to use this triadic model, the same that we have. So the animals would do the same as the dogs. And in the controllable situation and uncontrollable, we have the recordings from prefrontal cortex and hippocampus. And we have this resistant rats. And after the stimulation, we can see that there was an increase in the theta rhythmic. And in the helplessness animals, they don't show that. So this is, here we can see the pointer here. The resistant animals, they have a huge signature that either in the frontal cortex, in the hippocampus, that helpless animals, they don't have. And also, there is a coupling between resistant animals during the stress, and there are also there is a coupling between even animals that are resistant and receiving inescapable shocks. Means that we have all types of EEG patterns that we could classificate the three conditions: the low stress, controllable situation, and patterns that predict resistant animals. So just looking at this hypothesis, the summary, we have this control over this situation. It is learned by a higher level, and this situation is then we have in the controllable situation the prefrontal cortex projecting and controlling through GABAergic transmission the serotonergic neurons in the dorsal rapid nucleus, and we have signatures that could discriminate that. So coming back to the Arvid Carson lecture in 2000, he mentioned that and predict 20 years ago that here the new imaging techniques in conjunction with advanced computer-dependent statistics involving pattern recognition derived from a wealth of data with great complexity will probably prove extremely useful for very much help to bridge the gap between animal and observations in humans. And if nothing else, such a process will help to reveal the enormous wealth of our present ignorance about the human brain. And these things have been recently using the same approaches. This is a study from UCSF in San Francisco, that they use depth electrodes in patients with pharmacoresistant epilepsy, and they were able to classify individuals with depression with 89% of accuracy. So this is leading to use prefrontal cortex theta rhythmic as a promising therapy for treatment resistant depression. And so we see that these things are coming in several news. And here in São Paulo, the group of André Brunoni, they just established, I'm just finishing, established a non-invasive facility to use as in depressive patients, and we are collaborating with them, and to try to use that non-invasive techniques and to apply for epileptic patients. So just to thank FAPESP and also Danilo, who was the student who did this research. And also thank Professor Zago, that when he was a Dean of Research 13 years ago, or actually 11 years ago, he announced the You Speak Knowledge of Olympics that was supposed to aim to stimulate creativity and encourage people to present proposals that would, as a group, advance in technology. And it was a very small budget, $2,000, but it was the first budget that we bought the first viral vectors to study optogenetics. So thank you for your attention. So I think we are going to go to the next speaker. So Diego Rovaris, it's a pleasure to invite him, Diego, to give a talk. He is an assistant professor in the Department of Physiology and Biophysics at the Institute of Biomedical Science at USP. And he is the chair of the Adult ADHD Brazilian Network and the ADHD Working Group of the Latin American Genomics Consortium. Please, Diego.
Okay, good morning, everyone. Thank you for the introduction. As a young investigator, it's a big pleasure to be here in such an important event. Today, I will present you my group's projects on the molecular psychiatry of ADHD, attention deficit hyperactivity disorder. And I will start with a very brief introduction about ADHD, which is a neurodevelopmental disorder characterized by executive dysfunction due to inattention and or hyperactivity and impulsivity symptoms. ADHD presents a high prevalence in the general population and is linked to substantial economic burden across countries. And the most important fact related to this presentation is that ADHD presents one of the highest heritability estimates in neuropsychiatry, almost 8% in all age groups. So given the high biological component of ADHD, genomics became an important tool in the search of the genes underlying the ADHD, and the integration of genomics with other omics is essential for uncover not only disease pathways, but also to develop improve clinical classification and develop approaches for diagnosis and prediction of the disorder and more precise interventions. In this context, genome-wide association studies are good approach, and GWAS revolutionized the understanding of the genetic architecture of complex human traits. Through this approach, we can scan the genome of many people and cross the variability with a given phenotype. So how GWAS findings have contributed to molecular psychiatry in ADHD? The first outcome is gene discovery. We are working together with more than 200 researchers across the globe. The idea is to map the genes involved with ADHD. The latest meta-analysis of our consortia found 76 risk genes enriched among genes expressed in early brain development, aligning with the neurodevelopmental nature of ADHD. And the associated genes also enrich in several brain-specific neuronal subtypes and dopaminergic neurons. Another outcome is to understand the shared biology. So genomically, ADHD shares biology with autism, depression, smoking behaviors, insomnia, schizophrenia, obesity, and ADHD is genomically negative correlated with educational attainment and a variable called age of first birth that is actually a proxy for externalizing behaviors. Another outcome is to understand the clinical trajectories of ADHD. We are following up a cohort of adult patients with ADHD, and in our later studies, it was a 13-year follow-up study. We showed that the genetic liability to depression increased the probability of a stable trajectory of the disorder. And finally, risk prediction is an issue that we are studying. The polygenic score for ADHD do predict ADHD in independent samples, and as we can see in this graph, individuals that are in the highest deciles present a huge risk to develop or to be classified as presenting with ADHD. However, the effects of polygenic scores are more evident in European individuals because the discovery data sets are primarily European, so we have Eurocentric bias that is very detrimental not only for Latin America but also for high income countries. So if you keep doing this, actually this will exacerbate health disparities, so we need to change the things here. So what we are doing in this regard, I'm originally from Rio Grande do Sul state, but now I'm based in São Paulo. I'm the scientific coordinator of the adult Brazilian ADHD network. This network is a huge collaboration between these two Brazilian states. I'm also co-chair of the ADHD working group in the Latin American Genomics Consortium. That is a new consortium that will probably change molecular psychiatry in Latin America. And at the ICB USP, I'm the coordinator of the Laboratory of Physiological Genomics of Mental Health. I'm leading two major projects at Physiogen Lab. one of them is funded by FAPESP, and we are exploring the biological connection between ADHD and major depressive disorders in contrasting environments. And the second project is a full R1 funded by the National Institute of Mental Health, and we are looking at the connection between ADHD and educational attainment in contrasting environments. The major goal of the first project is to search for molecular and environmental signatures related to the comorbidity between ADHD and depression, or specific of each disorder. And the main tools we plan to use here are epigenomics and genomics and epigenomics. Of course, the biological connection between ADHD and educational underachievement. The main idea is to test if it's possible to translate Eurocentric ADHD and educational attainment polygenic scores in our contrasting environment. So we have a couple of questions to answer, and one of them is how do the European polygenic scores for ADHD and educational attainment behave in Euro-Brazilians living in different social contexts, or do the ADHD and educational attainment polygenic scores predict educational attainment related phenotypes in admixed Brazilians? We are part of the Ancestral Populations Network. In this consortium, we are Connect ADHD. This is a new and really nice consortium. Currently, APN includes 7 projects. We were approved a few months ago. There are more than 25 international and USA-based sites, more than 100 researchers working together to collect and harmonize thousands and thousands and thousands of individuals to revolutionize molecular psychiatry across the globe. And these projects present some shared goals. So our major goal is to compile a large collection of extensively characterized adult ADHD cases and controls at the sociodemographic, clinical, and biological levels, and obviously to make the ADHD research more diverse. Brazil is highly heterogeneous in terms of ancestry. There are some regions that present a high European component, while others present a more prominent African and Native American component. And this heterogeneous makeup is not unique to Brazil. For example, in the United States, more than 40% of the population is now non-European or non-white, and this is happening also in Europe due to immigration. So our plan for the next 3 years is to achieve 10,000 individuals. We are collecting São Paulo city, Pelotas, and Porto Alegre cities. We are working with data from 2 Pelotas city birth cohorts, and these samples are very good and well characterized over time, so it will be a nice source of populational controls that are really hard to collect. And for the cases, we developed an online platform in this platform called Otus, and through this platform we have a volunteer interface and a clinician interface. So in the volunteer interface, there are some modules that screen for comorbidities, other neurodevelopmental disorders, disease and health outcomes, life events, etc. And we developed a really nice monitoring system and an algorithm that automatically extract the data and create some reports. One of these reports goes direct to the clinicians. For example, in this graph, we can see a given volunteer compared to the ADHD and non-ADHD groups, and this volunteer present a severe ADHD. So with this report, our clinicians can optimize the clinical interview and confirm the clinical diagnosis of ADHD or not, and other psychiatric disorders as well. In just eight months of recruitment in São Paulo, we invited more than 3,000 individuals, almost 3,000 started our protocol, more than 2,000 complete the first phase, and almost 2,000 have already donated the biological sample, and these individuals were deeply phenotyped. Our biorepository is located at ICB USP, and we are storing blood samples, serum, plasma, isolated DNA. For a subsample, we are collecting feces for microbiome analysis. And here we have a huge scope for collaborations in parallel projects. We'll use a new method for genotyping called blended genome exome. And this method was developed by Broad Institute of Harvard and MIT, and we are participating in a working group called BGE working group in the Ancestral Populations Network. So the idea is to combine different samples with this method to perform large-scale meta-analysis across the globe. We have a modest computational infrastructure. This is something we are working on, and I hope FAPESP will help us. We are generating a huge amount of data, and you need to process and keep this data stored. And to finish, I want to show you preliminary results showing how polygenic scores can be a powerful tool in molecular psychiatry. We carried out a minimal sample size experiment. The outcome was educational attainment. The independent variables were polygenic scores for educational attainment and socioeconomic status. We established 4 groups according to ADHD status and details of genetic ancestry. And we calculate 2 polygenic scores using 2 different discovery data sets to give the effect size to calculate the polygenic score in our sample. And with only 130 individuals, we got significant associations in, and we were able to recover the same variance explained these studies found in Europe. And when they use the 23andMe sample as discovery, this tool, this polygenic score can explain 20% of the variance of a complex trait that is an important social determinant of health. So when we look to the other groups, the effects are negligible, and we have a good tool, but only for specific subgroups. And if you don't increase diversity in the discovered samples, we will increase health disparities, and we need to change this. We are very collaborative and connected. As mentioned, we are members of LAGC, but we are also members of the Psychiatric Genomics Consortium. In Europe, we are part of Enigma, It To Be Nice. I'm part of the next generation of the European College of Neuropsychopharmacology Network, ADHD Across the Lifespan. And we are members of the International Multicentric Persistent ADHD Collaboration that was initially an European consortium, but there are Europeans living outside Europe, so Brazil and United States are part of this consortium. There is space for France here, so we can talk about it. And I want to thank my team at Physiogen Lab and my Brazilian collaborators from the other universities involved. Thank you for your attention, and FAPESP for the invitation.
Thank you, Diego, for a very interesting presentation. So our next speaker is, I'm not sure if I will, Rahul Gaurav. So Rahul Gaurav has a degree in electrical engineering, Master of Science in Electronics Engineering, and holds a doctoral degree in Neuroscience and Neuroimaging from Sorbonne University. And so he has an outstanding multidisciplinary background. It's fantastic. And he works with movement investigation and therapeutics teams and the Center of Neuroimaging Research, CENUR, at the Paris Brain Institute. So it's a pleasure to have you here.
Thank you. Thank you very much for the kind introduction, and thank you, FAPESP, for the invitation. Bonjour, and good morning, almost good afternoon. So a lot of interesting discussions we had today on various areas of the brain. Particularly, you talked about prefrontal cortex and a bit of brainstem. So I would like to take you a bit deeper into the brainstem, particularly in the areas of basal ganglia, of substantia nigra, locus coeruleus, a bit in the tiny nuclei, to see what kind of people are at risk of developing neurodegenerative disorders, particularly the threat we are facing with aging population in all over the world. We have a very documented survey on the West, but it's also quite prevalent in Asia, in Latin America, with the aging population. So we'll see in my talk how, with these brain changes, using the biomarker studies, we can understand which kind of people develop Parkinson's disease. So a bit of insight about, let me fix the mic properly. So a bit of insight about our center. So I'm based almost a decade at the La Pitié-Salpêtrière Hospital. So this is Paris Brain Institute inside the heart of Salpêtrière, and we are affiliated with the Sorbonne University of Paris. And it's around 25 research teams there on various multidisciplinary topics, and around 11 core facility platforms as well. So you can see how it looks like. It's quite a new building, like around 15, 16 years ago. And Pitié-Salpêtrière is already quite known to many of you. So my team, as you mentioned already, is movement disorder. It's quite multidisciplinary, tackling various aspects of movement disorder. It's headed by Professor Bidaye and Professor Lerisi. And I'm largely associated with the imaging side as well. It's my second affiliation, which is Center for Neuroimaging Research. So it's a core facility comprising various facilities, not just human MRI, where I'm associated. We also have animal facility, PET MRI, etc. And just recently, like almost 10 days ago, earlier we used to have 3 Tesla MRI. Just like 10 days ago, we had a 7 Tesla, like really strong advanced technology of around 20 ton of weight of magnet being installed at the Paris Brain Institute. It's quite difficult and a lot of noise near my office. So let's start about the background of the study, what we are going to talk about. So often there have been many neuroprotection studies around, particularly focusing on neurodegenerative disorder, but they often fail. And why they fail is because not all the factors are taken into account. For example, if Parkinson's disease patient is taken into account, it shows tremors, but often we don't take into account the finger taps. For example, the finger taps could come from arthritis or other issues. This is just an example from a neurodegenerative disorder. So you could have similar other factors which create a sort of imperfect clinical trial, like a noisy trial. So this, how to tackle this? This brings us to the big question of biomarkers, particularly neuroimaging biomarkers. So biomarker can serve really as an early sign for your health, like how the disease can evolve, who can catch the disease, and what kind of population we can study using biomarker. So what is my focus is with using the MRI biomarkers that can help us to understand disease diagnosis actually at an early stage, and monitoring the progression of the disease using, for example, the brain changes what we will have, and could also analyze the disease modifying treatments if it is possible somehow. So let's do a brief insight on what is Parkinsonism. Like, I'm focusing more on idiopathic Parkinson's disease. As you can see in the animation, it's famous boxing legend, Muhammad Ali. He had a young onset Parkinson's disease. So normally when we talk about Parkinson's disease, neuroscientifically speaking, we have two targets particularly. First is the substantia nigra neurodegeneration, which is the dopaminergic neurons going down, and the iron augmentation in the substantia nigra going up. So these are the two hallmarks which we are trying to understand. So although this is quite sporadic Parkinson's disease, however, there are many other factors also playing, like aging, I already mentioned, and there are factors of environment and also of genetics which can play in Parkinson's disease, like what kind of people who would develop Parkinsonism. So there have been several reports actually recently saying the increase in aging population, particularly from the United Nations reports, that how with the aging population, we will see more and more Alzheimer and Parkinson's disease. You can see from this animation here that how there has been an increase in around 81% since 2000, and like causing around 300,000 deaths, increase of 100% since 2000. It's like an alarming situation for the development of neurodegenerative disorder throughout the world. So as mentioned, that makes us think to develop really accurate biomarker for the disease progression or for other things to tackle. So let's see who are these people who are at risk of developing Parkinson's disease. So recently we also learned in literature about some asymptomatic people not showing any symptom whatsoever of Parkinson's disease, but having this LRRK2 mutation. So there's a very interesting study coming out from East Asia where they show how it can multiply the chance, like almost twofold, of having Parkinson's disease in future. So for example, having these kind of subjects who are at risk of developing Parkinson's disease, by understanding the neuroimaging changes, we can study the brain changes in these asymptomatic genetic mutation carriers. So the second one for prodromal stage is also very interesting, because the second one, this is called REM sleep behavior disorder. So almost 80% of these people over a decade or so develop, like 18–19%, they develop Parkinson's disease. So this is something very interesting to understand, that what is happening with these people. Like normally the features they have is characterized by nocturnal violence. They are super violent when they are sleeping, and it's often their partners who report that they have this disease. So it's also sort of underreported disease, but they have a large chance of developing Parkinson's disease. So at Salpêtrière, we are one of the big centers for sleep disorder under Professor Arnold. So we get most of the REM disorder patients from there to scan at our MRI center. All good. So, oh, no, it's not good. Tudo bem. So, coming back to the REM sleep behavior disorder, I think I lost it here. All good, all good. Tudo bem. So for this, maybe it will be fun to see here. It was Murphy's Law. Nobody had this. Okay, I'll try to touch less. So we saw what is idiopathic Parkinson's disease. We saw the prodromal stage, like what kind of people could develop Parkinson's disease. So now let's try to understand the neurodegeneration at the level of brain, how we can understand the changes. So we'll try to see the hallmark, which I mentioned already. The first hallmark, what I said, was substantia nigra neurodegeneration. So that is the dopaminergic neurons going down. So dopaminergic neurons, they contain this pigmentation called neuromelanin. And using a certain technique of MRI called neuromelanin sensitive MRI technique, you could see these changes. So for example, if you go in these images, you could see here that the normal people, they start to lose here, what you can see clearly, the substantia nigra here. So neuromelanin starts to get lost in Parkinson's disease patient with respect to the healthy volunteer. So normally we could try to understand for these people also who are developing Parkinson's disease, and we could somehow think that they could lie in between the healthy volunteer and the Parkinson's disease patient, because they start to already lose the neuromelanin pigmentation somehow. So this is like a very strong technique which has been quite around in last decades, because it gives like a stronger surrogate marker for the Parkinson's disease development. And interestingly, the things are not that simple. So these are the people who are developing Parkinson's disease, but neuromelanin, as you can see in this plot, is the signal intensity of neuromelanin. It already, at the age of 4, it goes up, and at certain point, around 60 years or so, it starts to come down. So any person who's aging is losing neuromelanin anyway. And then you have this Parkinson's disease patient nearby who are losing at a faster rate. So we have to really take this also into account, how it's happening. And it's not clearly known what triggers the neuromelanin decrease. So second part is the RN augmentation in the substantia nigra. So we have other techniques of MRI called quantitative MRI techniques. This is very famous, called QSM, quantitative susceptibility mapping. Using QSM technique, one can quantify the RN augmentation in the substantia nigra as well. The third one is very interesting and quite common in clinical setting. What happens here? So if you see this bird, the swallowtail sign, so what happened in these kind of imaging, so this is high resolution T2 star or susceptibility weighted imaging. So you often see this in radiological practice when they have a doubt of Parkinsonism, let's say multiple system atrophy and others, versus being a healthy control. Normally the neurologist is doing the consultation, but if they come to a radiologist, they could see this swallowtail sign and compare that whether the swallowtail sign is staying or disappearing. Because literature also shows that there is a diagnosis accuracy of above 90% if it starts to disappear. So let's see this image where I have shown swallowtail present. It's not very clear, but you could see somehow the presence of swallowtail. And in this guy, the swallowtail has disappeared. That means there is a chance of Parkinsonism developing. So this is also a very, very strong marker. So we could think about developing a technology to quantify these things really precisely, which could answer a deeper question about neurodegenerative disorder. And the third one is a very, very difficult region to segment and quantify. This is called locus coeruleus, and completely locus coeruleus subcoeruleus. So in the MRI, we use the same technique of neuromelanin sensitive imaging, because locus coeruleus also has neuromelanin pigmentation, just like the substantia nigra. Just the difference is, there it is dopaminergic neuron, and here it's in noradrenergic neurons. Tiny bit, not like substantia nigra, but it's still. So here what you see is healthy volunteer having these 2 spots. And when you compare with the REM sleep behavior disorder patient, the locus coeruleus disappears. So you do not have left or right of locus coeruleus. So it's quite known that there is an implication of REM sleep in these subjects in the REM sleep behavior disorder, and the locus coeruleus starts to degenerate in this. So let's see now the methods, like what kind of techniques we could use to quantify using non-invasive methods, all these regions, to learn more about people at risk. So giving you a brief insight about MRI technology, if I think most of you are familiar with, but still, it's a non-invasive medical imaging technique that records changing field. You can see in this cartoon animation how it happens, that a person goes inside the MRI, it's fully non-invasive, and the experts could see the changes happening and can give different kind of images based on the pulse sequence. So you see also with this slice, how it would show using axial, sagittal, or coronal view. So it's my picture there in the 3 Tesla, which we had in the past, and now we'll have a better picture with the new 7 Tesla MRI. Murphy's Law is back. I think when I'm not touching, it disappears. All right, to the main. So coming back to the MRI technique, let's see the sequences, what we have. So often we have various kinds of T1, T2 maps, but in this talk, I'm largely focusing on neuromelanin imaging type, and you saw a bit about susceptibility weighted imaging as well, and largely RN sensitive quantitative MRI to quantify the RN sensitive changes as well. So the target in these studies are rather to accurately quantify the regions, like automatically having a non-invasive idea of how we can know more about these quantitative changes in the brain, so that we can see the progression or have the diagnosis accuracy in tracking using the MRI. So what we use here, we use a largely convolutional neural network. So what happens here, as you see in the animation, it is CNN is largely inspired from the human visual cortex, not I think I have too many animations. Maybe that's why. Can't handle. Very good. Okay, so it's moving. So the whole idea of deep learning is to design a sort of algorithm which could imitate intelligent human behavior. So in neuroimaging cases, what we do is very similar to this example, which I have taken from ChestNet. What they were trying to do to classify the diseases on the basis of various trainings they were having. Okay, this is what I was doing, and it got stuck. Okay, good. Okay, so yes, so that's the whole idea of deep learning. So we are trying to automatically quantify based on the training data what we have, as you saw in this animation. So what happens in these cases? You have a very simple example of, let's say, this tumor. So you just feed in the image in the model. You have to have a sophisticated model trained very strongly, and eventually it could find the tumor, for example. That's basically what we do in our case using units. So it passes through all the input layer and hidden layer, and eventually gives you the output very similar to what you have in the ground truth. So let's see the example of neuromelanin sensitive imaging, how it tries to quantify the region of substantia nigra, very similar to what a radiologist could do. So here you could see in this example, I think it's about animations, because I didn't realize I overloaded it. Yeah. So all right, so what you could see here in this example, in yellow is how the clinician is segmenting the region, and in red you see is automatically done using artificial intelligence technique, using deep learning. So it does a sort of 85% of similarity using the similarity index of dyes, etc. So it's trained on 60 images. So the basic idea is in MRI studies, you do not have huge data set. So 60, 100, 300 is already huge. So using these models, what we do, we try to use data augmentation techniques. That means when you have 60 images, 60 subjects, the model tries to flip it, create the left side as a right side, create mirrors, and eventually make the 60 images as, let's say, 6,000. And in this way increases in an artificial way the training data set, and it makes the model rather strong. So let's see how it would look. Like you have an MRI image of neuromelanin sensitive, and you just throw it in the model and what it does. So this is a very direct example of you send the image and it scans the entire slice up until the end and tries to identify the substantia nigra, and eventually gives you the volume and signal intensity and all for this, and which leads to compare. So recently we tried to use the similar model in the difficult region like locus coeruleus as well. So locus coeruleus you saw is very tiny, so understanding there is rather difficult. So this is how it looks like if you see these two tiny left and right over there in locus coeruleus. So this is without the region of interest, and this is the one done by radiologists. So even by radiologists, you see it's quite difficult. So we tried to train the model similarly using around 50-60 training data set, similar UNET model, and this is how you see the result. This is the one done using AI, and it all the time replicates with 85% of similarity or so. So this is a healthy volunteer where you have locus coeruleus, but when it starts to disappear, there is neurodegeneration, it becomes quite difficult. So let's see, this is an example of a patient with REM sleep behavior disorder, IRBD. So you see locus coeruleus is already not visible. It's very difficult to quantify. And you see how radiologists could do in yellow. Quite okayish, but it's still difficult for human eyes. And the AI would replicate somehow like this. Again, this was group-wise. When you compare, it gives you around 85% or so. So this was the model which we published a few years ago, the whole idea. And we also further compared the Parkinson's disease, IRBD, and healthy volunteer. So what you see here in the model, so you see the healthy volunteer is on the top, and it starts to go down the volume of the substantia nigra identified using deep learning. So the IRBD, as you see, stays in the middle. That means these are the people at the risk of developing Parkinson's disease. So even when they are at risk of developing Parkinson's disease, the neurodegeneration has somehow started. So this is a very interesting point we are trying to understand, that even when they are at risk, you are seeing these changes. So in the images, you could see how it looks like healthy controls. They have a lot of substantia nigra neuromelanin, and it goes down at the prodromal stage, and furthermore at Parkinson's. So these are Parkinson's disease of around 1.5 years or so. Further, we were trying to understand this biomarker, whether it can be useful for progression or not. And progression, we saw similar changes. You see here, actually healthy volunteers and early PD, these are the same group of around 1.5 years. And in progression, it's around 9.5 years or so. So you see here, they contain lots of substantia nigra neuromelanin. And again, it goes down at 1.5. And you see 9.5 years, it's almost disappearing, the substantia nigra. Similar, the plot is showing the similar change, the diminution at a faster rate for the progressing data set of Parkinson's disease. Further, we try to enter inside the substantia nigra to understand the subregional changes, because earlier we were trying to understand as a whole how it is changing. So here what you could see, that how the healthy volunteers and IRBD, always it stays in the middle. And for the progression, you see the changes happening at the inferior side. You could see in the substantia nigra, the last picture. So what it shows that the postoventral substantia nigra has more changes happening. So there the drop is starting to happen. So another part interesting was about the genetic mutation carriers, which are very, very difficult to find actually. So recently we collaborated with teams in Asia to identify these people not having any sign of Parkinson's disease whatsoever, but having LRRK2 mutations. So we collaborated with a team in Beijing with not many subjects because we needed to have MRI scans done in a similar way using neuromelanin imaging type. And we found the pattern you can see here is quite similar to the prodromal stage of IRBD. So Parkinson's disease somehow goes down, healthy stays high, and your asymptomatic LRRK2 carriers stays somewhere in the middle. Further, we were also trying to understand now with the locus coeruleus how the neurodegeneration is happening. So not just taking Parkinson's disease, taking Parkinson's with and without REM sleep behavior disorder, and having some more Parkinsonism like multiple system atrophy, progressive supranuclear palsy, and the isolated REM sleep behavior disorder, and comparing with the healthy volunteer. So what we found here, the locus coeruleus also, like you see the degeneration happening here, and more prominent in the synucleinopathy, because REM sleep behavior disorder patients are known to convert into Parkinsonism classically and less into PSP. That's why you see more, it's not many subjects in multiple system atrophy, but it shows the most changes. And the last study was on finding the iron augmentation in Parkinsonism. So this is, we are collaborating largely with neurologists, radiologists, and many physicists and engineers, because this is a big study. We are trying to have 3 visits here. Like people have been followed from 2013, 2014 or so, and every 2 years. And now we are trying to understand in this novel study how iron is augmenting over the years. Does it follow the similar pattern? What you see neuromelanin drop, is iron going in a similar way or not? Second target, what we did, rather than just seeing whole substantia nigra, we tried to enter the subregions. Like here you're saying is with the color coding, you could see the upper and lower part, like anterior, posterior of the dorsal and ventral slices. So here what we found in this study, the changes are happening in the postoventral, like the lowest part you saw in the substantia nigra for neuromelanin. In a similar way, that lowest part contains the most iron as well. So iron keeps on increasing as well. So that is the first baseline part where you see the healthy volunteers carry a lot of iron, and the REM sleep behavior disorder going up and Parkinson's disease going further up. And longitudinally, you see similar patterns slowly, slowly. Like you see at 0 years, after 6, 7, 8 years, it starts to go up. Although seemingly the speed is not like what you see the decrease of neuromelanin. So we are still working on this research to understand how the Parkinson's disease shows the increase over the 3 visits over the years. So the final point about summing up what you saw. So the conclusion, how neuroimaging biomarkers could be very strong marker and a very strongly non-invasive ones to not just understand the diagnosis accuracy, but also progression marker, and possibly could be the first step of implementation in clinical practice. However, there is a large part which is not so studied in these areas, which is largely about comprising more and more clinical, like motor, non-motor sites, and also genetics, which people mentioned already about. So having sort of a combined marker, like a genet MRI, which could unravel the understanding of Parkinson's genetics outside the European population, because these are all European populations we are studying. So in this way, we could identify all other regions, like where it's happening, the changes with respect to the risk of developing Parkinson's disease and Parkinsonism in general. And a quick shout out to my team. So it's like like a decade of studies I cited. So lots of thanks to all of them, and muito obrigado to all of you for the kind invitation. Thank you.
So the next speaker, it's a pleasure to have Guilherme Polanski here. He is graduated in medicine from Federal University of Rio Grande do Sul. at residency and a master and doctoral degree, also in URGS, and he has a postdoctoral at King's College, and he's associate professor of the Department of Psychiatry at USP here in São Paulo, and head of the Child and Adolescent Psychiatry Service at the Institute of Psychiatry at Hospital das Clínicas of USP. So it's a pleasure to have you.
Thank you. Good morning. Thank you again for the kind invitation. I understand that I am the last barrier for you to have lunch. So I'll give you a brief overview of one important area of research of our group. So I'm a clinical scientist, and our research group is clinically oriented, even in populational samples or clinical samples. And these projects that I will show you briefly here are most of them are the result of my thematic project, FAPESP project, that was just finished 2 months ago. So neurodevelopmental disorders is the main area, the main disorders that are the focus of our research. As Diego mentioned to you before, these are conceptualized as disorders that result from deviations of typical brain development, and the ADHD, autism, intellectual disability, motor, language, and learning disorders are some of these disorders. The causal and pathological process are, of course, in place years before the first symptoms are expressed and the diagnosis is established. So this is one area of our interest, to understand the first signs, the emergence of these neurodevelopmental disorders, especially ADHD. ADHD is and the disorders are highly heritable. Genes are shared between the disorders with other mental disorders and with cognitive functions. And the environmental risk factors that so far have been identified, they are concentrated early in life, prenatal and perinatal periods. In parallel to that, we understand that poverty is one of the most strongest risk factor for mental disorders. Poverty is correlated to violence, is correlated to undernutrition, is correlated and with all sorts of risk factors that you can imagine that will impact negatively the development of a child. And unfortunately, in Brazil and in several Latin American countries, there is a huge, as you know, economic disparity, and there is still lots of children, millions of children living in poverty. So the project that I will show you briefly here are trying to address this overarching research goal to study developmental trajectories of neurodevelopmental disorders and to test early interventions with the aim, these interventions with the aim to attenuate the expression of cognitive problems that will precede later neurodevelopmental disorders. So we have been studying the first or the most early expressions of ADHD early in development. This is a meta-analysis that basically shows that the same precursors of autism are also the precursors of ADHD. In this regard, we have proposed, along with other colleagues from a consortium on early ADHD, a developmental cascade that starts, of course, with genetic susceptibility. There are inflammatory processes that have been the focus of interest of several research groups, prematurity and low birth weight, early sensory and motor processing problems, sleep problems, and early temperaments, along with quality of parental care, are those characteristics that precede ADHD. despite the fact that ADHD is strongly related to autism, we have been studying what differentiates them. And this study from our research group using network analysis in a sample of more than 10,000 adolescents in Brazil shows that there are specific symptoms that I'm not going into details here. There are specific symptoms of ADHD and autism that are strongly related, and there are behavioral and cognitive explanations for them that are interesting to pursue. In this, also in this area, this is a longitudinal study. Actually, we investigated 2 samples from the UK, more than about 10,000 individuals followed up to 20 years. And in these 2 samples, we documented that that depression, which is a comorbidity that has been cited and studied by our colleagues, that depression that emerges during the development of children with neurodevelopmental disorders are associated with psychosocial risk and exposures that these children are exposed, and not directly with ADHD and autism symptoms. Focusing also in observational studies, this is a project that is not funded yet by FAPESP. This is a project with several colleagues from USP. Patricia Braga is here, and she is one of the PIs of this cohort, that we are evaluating 555 infants. They were included at age 3 months, and they are being assessed 5 times until the age of 3 years. And the aim here is to understand the early precursors of executive functions, which is one of the hallmarks of ADHD. So we are studying these children in a variety of aspects. We are studying collecting EEG. The environment of these children and the quality of parental care is assessed. Cognition and behavior with standard protocols, genetics and epigenetics, and microbiome. So we have data from multiple levels of interest, and we aim to, as we conclude the study, to understand the precursors of early executive functions at age 3. There are probably 1 or 2 publications of this study. This is an ongoing study, but this is some analysis that, several analyses that we've been conducting, and this here, for example, shows the trajectory of the maturation of specific frequencies as measured by EEG. And here, for example, the precursors or the predictors of regulation problems. So here, for example, the PRS for bipolar disorder is associated with problems in regulation early at age 2 years. And cognition at age 2 years is also in this model associated or predicted by PRS of Alzheimer and schizophrenia. So these are some of the observational studies, and there is another area of intervention studies, although the specific pathway that leads to neurodevelopmental disorders in terms of mechanisms are not completely elucidated. The knowledge we have so far in terms of risk factors, we understand there is enough to propose interventions. So I'll show you 2 interventions in populational samples. This is an intervention focused on problems of social communication. And this has been developed for children with autism. But here we are studying this intervention for in the trial 1 for children who are living in extreme poverty, who are already presenting social communication problems in the first 2 years of life. They don't have autism, so they are receiving this intervention that is aiming to prevent or to attenuate the expression of later problems that these children will have. Professor Alicia from the Department of Public Health is the PI of this project. And the second project, which is the Jovem Pesquisador of Lizzie Shepard, who is now at the Psychology Institute. She's a faculty, just has been approved there, studying children with high risk for autism and ADHD. Another project also focused on populational samples is a home visiting intervention program for adolescents who are pregnant for the first time. They are 14 to 19 years old and they are pregnant. They live in extreme poverty. So this intervention consists of nurses going every week, visiting these families and later the children, starting in the first trimester of the pregnancy, and we finished by the second year of the life of children. And we conducted 2 trials and also assessed these children in a variety of ways, measures that are similar to those that I have already mentioned. So, for example, in this study, we showed that this intervention improved. I will not dare to touch in this thing. So this improved emotional and verbal responsivity of the mothers in terms of their children, which is essential, and it is one of the probable mechanisms by which the intervention will work. We showed that on a specific measures of EEG that is related to social communication, using an experimental paradigm, the mother face and the stranger, we demonstrated that those children who received and whose mothers also received the intervention, they have larger amplitude of these specific measures, meaning a better functioning of the process, and this is associated with later communication, social communication skills. So this is a specific measure improved in those who received the intervention, and it is later associated with better communication skills in these children. We also showed that children who received the intervention and their mother had a better relationship, and this is measured here by observation of attachment. So these were blind measures, and what happened is that children who were born to adolescents' mother are described to have about 60% of indiscriminative or insecure attachment. And what we showed is that those who, and this is also demonstrated in those who did not receive the intervention, but those who received the intervention had improved, and just 36% of them, which is the general population rate described in other studies, had insecure attachment. And finally, at age 2 years, these children had better verbal expression measures. This study is now being, this intervention is now being implemented in two cities. So there is a special program of FAPESP called CISME. Euripides Miguel is the PI, and we are now implementing this intervention in 2 cities funded by the CISME FAPESP. The last intervention that I'm going to show you is in a clinical sample. So ADHD can be diagnosed and is diagnosed at preschool age. I'm not going into details, but it is not clear what is the best treatment for preschool children with ADHD. And we conducted the most well conducted, and I'm sorry, I am the PI, but I have to say, but study in the literature so far comparing behavioral parent training with methylphenidate and with control groups that assure us that the individuals who received the behavioral parent training are blind to this intervention. And we showed an improvement in ADHD symptoms for those who received methylphenidate and global functioning for those who received behavioral parent training. And in the secondary analysis for our secondary outcomes, we showed the specificity of interventions. So those who received methylphenidate improved in terms of cognitive measures, and those who received behavioral parent training improved in irritability, which is a big problem in these children. I'd like to acknowledge my research group and thank you for your attention at this time.
Everybody must be hungry, but we have come to the speakers to just a couple of questions. Oh. Questions? I have a question to Rahul. Rahul, I think that you showed very interesting images regarding the tiny structures like locus coeruleus and the substantia nigra for some of as a marker for Parkinson's disease. So, do you have an explanation or a histological or histopathological study to demonstrate in histology that we have iron deposit in the posterior region that you showed in the MRI studies? And also, there are some. Is there a protocol special? Or is it just a T1 with some? There is no contrast. So this is just a protocol, T1 protocol adapted to melanin and iron.
Thank you for the question. So first, picking up the second question. It's not a general T1 protocol. It's a specially dedicated protocol for T1 type weighted neuromelanin sensitive. So this one which I showed is Siemens Turbospin-Echo, like really dedicated one for the neuromelanin. And for the iron, these are quantitative. So we acquire, like there were phase angles, and experts of QSM actually, experts, they would prepare with the reconstruction of images. So there is a bit of procedure also involved. So that's one of the negative side for having big multi-centric studies. So you need to educate everybody how to have these kind of protocols and do reconstructions as well. So that was for the second. For the first part, for neuromelanin and iron and all, there have been studies validating, but really going deeper on having ex vivo studies. This is what is the idea for the collaboration which we have been talking about with Brazil, to go deeper with in comparison, like in situ comparisons for these studies. So that's the idea, like I have mentioned a couple of times, with the potential collaboration we might have with USP. It is in the same regard which we were talking as well. Yeah.
Do you have any idea how about the expression of receptors that may explain why iron is deposited there? I mean, for example, transferrin receptor that binds transfer load by iron. Is this upregulated as well? My question is whether, for example, venetoclax is a drug that can block iron uptake, right? So this can be also probably something that can be think about treatment in Parkinson. What's your think about?
Yeah, this is like a long debate which hasn't worked so far, any sort of blockade like that, as far as I know, for iron augmentation. But that could be possible, but I'm not sure how, because it's very tricky, you know, to know how iron is increasing as well, because it's happening at the same time somehow. But what is coming before is not really clear. We have tried to understand that iron augments and neuromelanin goes down in the same posterior part of substantia nigra. But how it's happening, what is triggering precisely, is not really clear.
Look for the transferrin receptor.
Yes.
Yeah, it should be interesting.
Yes, it could be. That's why we need more and more validation studies using other modalities and more multicentric studies to really understand the precision.
So Diego has a question to Guilherme.
Guilherme, yesterday I was talking with Patricia about methylation and epigenomics and the epic array. Are you considering to carry out the epigenetic analysis considering all time points? in your first study?
So, so Patricia is the responsible for this part of the project, and hopefully we'll find someone who would like to fund us.
Oh,
Maybe, maybe.
Yeah.
But definitely we are collecting biological samples, and this is a very special sample. So yes.
Yeah, it would be a very unique study in terms of epigenetics.
Yes, the whole challenge of this kind of studies, and for those who conduct longitudinal studies know is to actually to have these participants in the study over time. So hopefully we'll have these participants at age 7, at age 10, and then we'll be in a very good position to understand early risks and precursors of ADHD or other problems in general.
I have a question for both Guilherme and Diego. So, is there any changes in this longitudinal studies with the relating the use of gadgets, cellular phones, and the increase of ADHD as a environmental interference?
Yes, this is a big question all over the world because, of course, we are all using a lot of cell phones and our children as well. So the quality of evidence is really, really limited, but so far the best evidence shows a very small increase in dissatisfaction and depressive symptoms in specific ages, but not for ADHD. There are several small studies with 2,000, 3,000, 5,000 individuals, but we don't understand this is still enough to demonstrate this effect.
I have a quick question to Diego. You showed a very interesting network of consortiums where you showed Enigma. Enigma is one of its kind, combining like world-class neuroimaging data with genetics, which is not so existing. Could you enlighten us more about that? Like, I don't know how you integrate. Are you already working with Enigma?
Actually, our colleagues from Rio Grande do Sul are part of Enigma ADHD, and they are sharing neuroimaging data with genomic data. So they are carrying out meta-analysis in this regard, but I'm not working directly with them. But it's a huge consortium, and I think if you have data to join in, we can make the bridge.
Well,
Alexandre? So, if you're not having more questions, I invite you to have lunch. I don't know if there is some advisement. Okay. Thank you for your audience.
I have some comments here. 4 reasons for you not to leave FAPESP, for run away from FAPESP. First one is lunch, of course. We're having lunch, and we'll come back at 2 p.m. Second one is gonna take a picture, a group photo here up front, if you please help us out. Then I'll pass around during lunch some receipts for those who are being paid for FAPESP. So please be aware that we are collecting some signatures. And least and most importantly, we have a quick talk at the end of the day. It's not going to be too late, showing how FAPESP can fund research. Okay, so this is important. That's where the money is. So be back. So.